25 dalam acara Seminar mRNA Innovation to Implementation Integrating Science, Regulation, and Global Collaboration. Boleh berikan tepuk tangan yang sangat meriah Bapak Ibu untuk menambah semangat kita di siang hari ini. Distinguished participants, ladies and gentlemen, allow me to greet the dignitaries here with us and through the Zoom meeting. Honorable Your Excellency, Minister of Health of the Republic of Indonesia, Mr. Budi Gunadi Sadikin. Welcome, sir. Distinguished Director General of Pharmaceutical and Medical Devices, Mrs. Lucia Rizka Andalusia. Welcome, ma'am. Distinguished Vice President of External Innovation of CanSino Biologic Inc., Dr. Chenlin Sin. Welcome. Distinguished Associate Director General, International Vaccine Institute, Professor Susan Sastra Buddha. Welcome. Distinguished Director of Health Product Policy and Standard Department World Health Organization, Dr. Dus Dedit Mubang Nyizi. Welcome. Distinguished Program Leader, RNA Program, Coalition for Epidemic Preparedness Innovation, Martina Och, PhD. Distinguished Senior Official of the Ministry of Health of Republic of Indonesia and other related ministry and institution. Distinguished Speaker and also ladies and gentlemen, once again welcome to Seminar mRNA, Innovation to Implementation, Integrated Science, Regulation, and Global Collaboration. We'd like to extend our gratitude and appreciation for the presence in this wonderful occasion. Bapak Ibu yang kami hormati, saya mengaturkan rasa syukur dan penghargaan atas kehadiran Bapak Ibu sekalian pada acara kita di siang hari ini. Tentunya acara hari ini diselenggarakan oleh Direktorat Jenderal Kefarmasian dan Alat Kesehatan Kementerian Kesehatan Republik Indonesia. Dan senang sekali hari ini saya Bella Idarwan dan juga menjadi sebuah kehormatan untuk saya dapat memandu rangkaian acara yang sangat spesial di siang hari ini. Hadirin yang kami hormati, sebelum kita memasuki acara inti hari ini, dengan hormat kepada seluruh undangan, kami mohon berkenan untuk dapat berdiri sejenak. Kita akan bersama-sama menyanyikan lagu kebangsaan Indonesia Raya. Menyanyikan lagu kebangsaan Indonesia Raya. Hadirin, kami persilakan untuk duduk kembali. Hadirin yang kami hormati, selanjutnya dipersilahkan kepada kita semua Untuk dapat menundukkan kepala sejenak, seraya berdoa agar seminar hari ini berjalan dengan lancar, sukses dan tentunya membawa manfaat untuk kita semua. Berdoa sesuai agama dan kepercayaan masing-masing, berdoa dimulai. Berdoa selesai. Hadirin yang kami hormati, sebagai acara pembuka, marilah bersama-sama kita akan mendengarkan laporan yang akan disampaikan oleh Direktur Jenderal Kefarmasian dan Alat Kesehatan. Yang terhormat Ibu Lucia Rizka Andalusia, kami silahkan.
Assalamualaikum warahmatullahi wabarakatuh. Yang saya hormati Pak Menteri Kesehatan yang sudah berkenan hadir pada siang hari ini, Bapak Ibu narasumber, Bapak Ibu perwakilan dari kementerian lembaga, dari perguruan tinggi, dari industri farmasi, dan juga saya yang penting lagi Pak Menteri, ada Direktur Riset LPDP Pak. Nah, kenapa saya bilang penting? Karena kita butuh anggaran riset dari beliau. Baik Bapak, kami akan melaporkan bahwa sesuai dengan arahan Bapak, kita mengharapkan bahwa penguasaan teknologi tidak eksklusif, tapi kita buat inklusif, di mana seluruh peneliti, baik itu dari perguruan tinggi, industri farmasi, lembaga riset maupun rumah sakit, memiliki kemampuan untuk melakukan riset dan pengembangan teknologi kesehatan dengan teknologi-teknologi yang baru. Secara khusus, teknologi mRNA ini adalah teknologi untuk mendevelop suatu vaksin dan produk biologi yang kita harapkan nanti tidak hanya untuk vaksin, tapi juga untuk produk-produk yang lain. Oleh karenanya, kita ingin agar inklusivitas ini terbentuk untuk seluruh Indonesia, meskipun di awalnya riset ini dikembangkan di pusat riset dan development Bio Farma industri vaksin kita. Tentunya ini nanti yang menikmati bukan hanya Bio Farma saja, tetapi seluruh riset dan development department di Indonesia ini. Next slide mungkin. Jadi pada kesempatan ini kenapa kok kita mengangkat teknologi mRNA? Karena teknologi mRNA ini adalah suatu fondasi, salah satu fondasi yang penting dalam kita memperkuat ketahanan kesehatan di Indonesia, khususnya adalah terkait dengan pandemic preparedness response. Sebagaimana kita ketahui ketika kita pandemi kemarin, kita mengalami suatu kondisi, kita membutuhkan perbekalan kesehatan, termasuk di dalamnya yang sangat penting adalah vaksin dalam jumlah yang banyak, dalam waktu yang cepat. Dan sebagaimana kita ketahui, teknologi mRNA ini adalah suatu teknologi cutting edge technology yang dapat memproduksi vaksin dengan cepat dalam waktu yang singkat, dan juga kita harapkan ini nanti menjadi suatu teknologi yang bisa kita gunakan dalam pengembangan produk-produk lain. Dalam dimensi National Health Security, tentunya kita menempatkan penguasaan teknologi mRNA ini sebagai komponen strategis, sehingga kita dapat merespons kondisi pandemi atau ancaman-ancaman kesehatan lain, dan secara mandiri kita bisa memproduksi di dalam negeri. Tentunya ini sesuai dengan pilar ketiga, yaitu ketahanan di bidang farmasi dan alat kesehatan. Dan kita mengharapkan suplai perbekalan kesehatan khususnya vaksin ini dapat terjaga kontinuitasnya dengan kita mampu memproduksi di dalam negeri. Penyiapan infrastruktur riset ini dan manufacturing penting untuk membangun kesiapsiagaan kita, dan tentunya tidak bisa dilakukan sendiri, ini harus dilakukan secara bekerja sama terintegrasi, baik itu lembaga riset, kemudian industri farmasi, dan yang paling penting juga pemerintah sebagai regulatornya, dan juga kita perlu kerjasama dengan international organization. Dalam hal ini pengembangan mRNA vaksin kita banyak mendapat support dari international organization untuk membangun kapasitas di dalam negeri, baik itu mulai mendapatkan lisensinya sampai kepada alih teknologi atau transfer teknologi dan juga support funding untuk alat-alat dalam hal ini untuk penelitiannya. Next slide. Nah, ini yang selalu disampaikan oleh Pak Menteri, penguasaan teknologi vaksin kita tentunya harus berkembang. Saat ini yang sudah ada dengan teknologi-teknologi vaksin yang lama ya, dengan konvensional teknologi, inactivated virus, live attenuated, kemudian juga protein subunit, dan virus-like particle yang sekarang kita kembangkan adalah viral vector dan mRNA. Teknologi vaksin ini akan memperkuat bagaimana produksi vaksin dan produk biologi lain tentunya nantinya. Jadi bukan hanya kapasitasnya dari industri vaksin 1 menjadi sekarang sudah 4, tapi juga penguatan teknologinya, kapabilitasnya. Karena ketika nanti kita akan bekerja sama, apakah itu untuk supply di Indonesia maupun global supply, pasti akan dilihat kemampuan teknologi kita di mana. Nah, dengan kita dapat memproduksi berbagai platform vaksin ini, harapannya ke depannya selain untuk ketahanan di dalam negeri, kita juga dapat berkontribusi di tataran global dalam penyediaan vaksin dan perbekalan kesehatan yang khususnya dengan teknologi-teknologi yang baru. Nah, Bapak, kami menyampaikan, next slide, roadmap ya perkembangan vaksin ketika tahun 2021, mungkin Bapak masih ingat ketika WHO memberikan kepada kita express of interest bahwa Indonesia ditunjuk sebagai WHO MPP mRNA Technology Program, dan ini sudah didukung juga oleh letter of support dari Kementerian Kesehatan sehingga Indonesia ditunjuk sebagai salah satu negara yang mendapatkan alih teknologi, transfer teknologi mRNA ini melalui WHO MPP. Itu di tahun 2021. Kemudian juga Bio Farma mendapatkan funding support untuk fasilitas produksi mRNA ini. Setelah itu berbagai kegiatan dilakukan, mulai dari training yang difasilitasi oleh WHO, kemudian pada tahun 2023 juga Bio Farma bekerja sama dengan CEPI, dari due diligence CEPI menunjuk Bio Farma sebagai Global South Collaboration untuk Vaksin Manufacturing Facility Network, dan juga penguasaan teknologi, di mana CEPI mempropos penguasaan teknologi pembuatan vaksin mRNA berbasis mRNA ini dengan alat yang kita kenal dengan kuantum. Nah, setelah itu mulailah dilakukan berbagai kegiatan penguasaan R&D dalam memproduksi vaksin dengan kuantum teknologi ini. Dari kuantum nanti akan juga hadir sebagai narasumber sehingga teman-teman yang lain dari industri farmasi lain, dari institusi riset juga bisa mengenal bagaimana teknologi kuantum tersebut, dan besok akan dilanjutkan dengan hands-on workshop ya, wet workshop di Bio Farma. Setelah Bio Farma berhasil untuk melakukan validasi dan engineering run untuk vaksin mRNA COVID-19, sudah sampai pada clinical lot fase 1 dan karakterisasi produk serta melakukan persiapan untuk engineering dengan skala yang lebih besar lagi. Kemudian juga telah mengembangkan seed mRNA MERS bersama Universitas Padjadjaran, oleh karena itu hari ini juga ada dari Universitas Padjadjaran yang juga mengembangkan plasmid DNA, serta kemudian juga pengembangan vaksin rabies yang juga sedang diuji cobakan dengan alat kuantum ini dan telah berhasil sampai pada produksi clinical lot. Nah, tentunya ini semua sekali lagi sebagaimana arahan Pak Menteri, kita mengharapkan penguasaan teknologi ini bisa tersebar bukan hanya di Bio Farma saja. Oleh karenanya pada kesempatan ini di merayakan Hari Kesehatan Nasional yang ke-61 kita melakukan workshop Kementerian Kesehatan bersama Bio Farma untuk diseminasi dan pengenalan teknologi mRNA dengan teknik yang baru, yang lebih cepat dan lebih praktis, efisien dengan kuantum ini. Kemudian juga tentunya kolaborasi dengan international organization sebagaimana tadi saya sampaikan, ini juga menghasilkan beberapa produk-produk yang nantinya akan merespon kondisi pandemi bukan hanya di Indonesia saja, tapi kita juga mensupport kondisi di global. Next slide. Bapak Ibu yang saya hormati, Pak Menteri, sebagaimana arahan Bapak bahwa nantinya teknologi mRNA ini bukan hanya untuk vaksin saja, karena ada potensi pengembangan produk lain, secara khusus adalah produk biologi, seperti pada produk-produk biologi untuk terapi kanker, imunomodulator, dan juga terapi-terapi yang lain yang menggunakan platform mRNA. Sementara untuk yang sudah ada dalam roadmap bersama CEPI dan bersama beberapa kolaborator lain, kita memprioritaskan untuk vaksin MERS, Lassa, dan Chikungunya, Nipah, serta disease X. Disease X ini nanti akan dibuat berdasarkan mapping patogen mapping yang akan dibuat bersama CEPI, prioritasnya untuk masing-masing daerah berbeda, masing-masing region berbeda, dan juga tergantung pada pola pandemi yang terjadi. Tapi dengan kita sudah memplot disease X ini, kita mempunyai kesiapsiagaan dalam merespon pandemi. Selanjutnya di ASEAN sendiri, karena kita telah mempunyai mapping terkait dengan penyakit yang sifatnya endemik di kawasan ASEAN, termasuk malaria, dengue, tuberkulosis, dan rabies, maka ini juga akan menjadi priority dalam R&D kita. Ini mungkin teman-teman yang di perguruan tinggi dan lembaga riset juga sebaiknya kita sama-sama memiliki pipeline dan roadmap yang sama. Tadi Pak Menteri sudah sampaikan bahwa Bio Farma mRNA Vaccine Facility ini bisa kita gunakan bersama-sama dalam bentuk kolaborasi, baik di perguruan tinggi, kemudian lembaga riset, serta industri vaksin yang lainnya. Dan kalau kita sama-sama memiliki suatu kekuatan ekosistem riset, akan sangat mudah bagi kita untuk propose funding untuk riset ini. Terutama kalau kita mau ke international organization, karena dia akan melihat kira-kira ekosistem risetnya mendukung enggak. Terutama juga nanti kalau sudah sampai di hilir, sampai di uji klinik, apakah tempat uji kliniknya mendukung atau tidak. Jadi ini yang kami siapkan Pak Menteri sesuai dengan arahan Bapak. Mudah-mudahan ekosistem pengembangan riset vaksin ini terus berjalan dan memberikan keluaran yang bermanfaat dan sesuai dengan apa yang kita cita-citakan untuk mendukung ketahanan kesehatan di Indonesia, khususnya ketahanan di bidang farmasi dan alat kesehatan. Demikian Bapak yang bisa kami sampaikan laporan terkait dengan pengembangan mRNA hari ini, dan 3 hari ke depan kami mengadakan workshop berupa materi pada hari ini dengan pembicara baik dari WHO, dari IVI, dari industri farmasi vaksin, kemudian juga peneliti-peneliti dalam hal biosecurity, kemudian pengembangan vaksin mRNA-nya sendiri, maupun pengalaman-pengalaman dari industri farmasi. Demikian Bapak, terima kasih laporan kami. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih banyak Ibu Rizka untuk laporannya. Kami persilakan untuk kembali ke tempat. Hadirin yang kami hormati, marilah bersama-sama kita saksikan perjalanan pengembangan vaksin di Indonesia sejak pandemi dalam tayangan video berikut ini. Selamat menyaksikan. Baiklah, hadirin yang kami hormati. Selanjutnya, marilah bersama-sama kita akan mendengarkan sambutan serta arahan yang akan disampaikan oleh Menteri Kesehatan Republik Indonesia. Kepada yang terhormat Bapak Budi Gunadi Sadikin, kami silakan.
Jadi, Ibu Rizka, ladies and gentlemen, good afternoon. I'm not scheduled to be here. Actually, I was scheduled to be at another meeting, but Ibu Rizka insists me to come because she want to show me something. Tapi, Bapak Ibu, I give you a little bit of background. After pandemic, I realized that we need research capability in the health sector. Otherwise, our population is at risk. Jadi, by the end of 2022, kita start thinking how we can design a health research ecosystem properly. Gitu ya. Satu yang we have the must be-nya adalah change the law. Itu sebabnya di August 2023, it needs about a year, we change drastically the law. Jadi the existing law, walaupun sekarang dituntut banyak di Mahkamah Konstitusi sama teman-temannya Dr. Nizar, sama teman-temannya yang lain, kita believe bahwa ini akan create an open architecture ecosystem so we can promote healthcare research. Dan setelah August 2023, kayak material transfer agreement, simplicity of recruitment process, itu semuanya disimplify. Kita build our clinical research center back in 2024 second semester, fourth quarter. Itu September, August 2024. So 1 year after the law, we started the National Clinical Research Center. The objective, our research will be different with BRIN. BRIN is more on the TRL, technological readiness level 1, 2, 3, 4, 5. Yeah. We will be at 7, 8, 9. And closer to the bedside. Kalau BRIN will be closer to the bedside. We will be closer to the bedside. Nah, saya start to learn other countries gitu. Like example, Malaysia, Singapore, itu has done clinical research much more than Indonesia. Coba next slide. This is one example yang I asked the inventor of Malaysia Clinical Research Center to share with us. Yeah. I learned they established the unit in 2007. By 2020, in 13 years, they achieved the target. I said to Indri, in 13 years, I will be retired. I don't know whether I still live or not. Gitu kan? Let's accelerate this time frame into less than 5 years. But you are a ridiculous person. You know, Indri, I'm a physicist. In physics, there is a famous physicist called Albert Einstein. And he is very famous because of his relativity theory. The essence of relativity theory, you are a twin, and your twins goes to Pluto and back to Earth at the speed of light, and then you meet again, and then your twins will look older than you. So the essence is time is relative. Is the Malaysian do it in 13 years? Well, we can do it in 5 years. Gitu. It doesn't mean that if Malaysian do it in 13 years, we have to be at 13 years. Because again, time is relative, especially from the eyes of a physicist. Gitu kan? Because it's based on scientific evidence that time is relative. So actually we can do it in 5 years, 4 years, and 3 years, depend on how fast we are in traveling comparable to the speed of light. Gitu kan? Theoretically, the science already confirmed bahwa time is relative. Jadi aku bilang, Indri, let's replicate what they have done in 4 years to 5 years. Gitu kan? And as a banker, not a physicist that become banker, I love numbers. Gitu. Coba go to see the numbers. Ya. Next slide. These are the numbers. Gitu kan? So do it in 13 years. Why don't we do it in 5 years? So in 5 years, your target is minimum 200 sponsored clinical research every year. Compared to 7 or 9 now, gitu kan? That's good. Ya? in PE, in finance, we love to talk about multiples. We talk about 30 multiples or 35 multiples. I love that multiples, from 7 to 200 a year. Then the revenue gitu kan, karena di sini ada Yuli dan Dr. Nizar, very proud. Pak, we get 200 billion, 300 billion. The revenue should be at least 300, 400 Malaysian ringgit million, 1.6 trillion per annum. Gitu kan? 1.6 trillion per annum rupiah sponsored clinical research, and then 200 sponsored clinical research per annum in the next, by the end of 2029, gitu kan, when I retire. That's a good target, healthy target, make us healthy. Of course, we have to prepare the platform, the ecosystem. The law is very important. The structure is very important, which is we have done it 1 year after the law was implemented, October 2024, and then we have to fill in with the people, and then we have to strengthen the lab. We have to strengthen also the ethical approval, make it the shortest as possible. If South Korea is the best, can do it in 4 weeks, let's aim for 2 weeks. Gitu kan? So that is let's give a clear ethical approval, not more than 2 weeks. No, not more than 2 weeks. We have to train the team also, the principal investigator. We have to have a series of principal investigator. It has to be clear that the company should deal with the CRC not directly to the principal investigator, because sometimes they don't understand how to write a legal documents, how to process, how to get resources, etc. And also sometimes it makes it difficult for the sponsor to deal with the principal investigator. And sometimes there are a lot of conflict, or the principal investigator want to keep it by themselves, want to get a journal for themselves, and then we don't get the best deal for the nations, for the country. So that's why it is something that we would like to improve. The target is simple: 200 sponsored clinical trial every year with the annual revenue 1.5, 1.6 trillion. And it's already proven coming in. The Gates Foundation sponsored our tuberculosis vaccine, clinical trial for 3. Itu berapa, Bruce? 25 million, 24 million. And the second vaccine will be, a clinical trial will also coming in. There is a long-lasting tuberculosis injections that we are looking to coming in, and also the clinical trial for 1 month tuberculosis medicine instead of 4 months and 6 months for both resistant TB and normal TB also will coming in. I just heard from PON, just got a tiny amount of clinical research funding, gitu kan, tiny amount, but they have GLP-1 drugs from Korea, 6 months or 4 months, and the patient is already coming in. The patient is already sitting in the bed, ready for injections and monitoring. That is level 1 clinical trial. So it start coming in. So hopefully more research are coming in. And from the Ministry of Health, we have 3 focus. One is biotech business, biotech in healthcare. Number 2 is artificial intelligence in healthcare. And number 3 is robotics in healthcare. So the biotechnology has been advancing quite fast. I've seen some of the research. RSCM has already issued some products and received some patent out of this. The artificial intelligence hackathon has been done. Several projects has been running. We are working with international companies. Hopefully, the first product will be launched very, very soon next year. Yeah? First and the second product I see on the pipeline soon will be launched based on AI. On the robotics, we start reviewing all the robots manufacturer that will work with us. Now everybody is coming to us. Before, the Intuitive doesn't want to sell the robot to us, but listening that this Ministry of Health is so ambitious, now they're coming in from India, from China, all the robotics company are coming in. Now we are reviewing which robots that we will buy will guarantee the technology transfer and the usage of robots in modern technology. So that is the overall research. But now back to mRNA as one of the component of biotechnology that I would like to push. And thank you for the vaccine manufacturer that, because it started in 2023 when I met with a very senior vice president from the Gates Foundation. He said, Budi, I know you are a very ambitious person. You want to get things done and you really want to get mRNA product. Why don't we give it to you? Since you are kind enough to put Ibu Rizka as the board of CEPI. No, I try to put Indonesian representative at the international organization board of Global Fund, board of Gavi, board of CEPI, board of PAT. So we put Indonesian there. Ibu Rizka sit at the board of CEPI, which CEPI was sponsored by the Gates Foundation. And I said, one of CEPI mission is when the pandemic happens, until the first vaccine available in the market will be 100 days. Only mRNA technology can do it. Only mRNA technology can do it. So that's why they're really pushing on the proliferations of mRNA technology. mRNA technology. Although, Ibu Rizka, in my view, I don't want to stop using mRNA technology on infectious disease vaccine. I want to move to another step, hopefully next year, to cancer vaccine. And then I move another step, if possible, to rare disease treatment. I know mRNA, because there is some rare disease that you don't have a certain type of protein. What I read, mRNA can be used to regenerate a certain protein that we don't have because of the rare disease conditions. Or maybe phase number 4, you can use not only the mRNA, but the lipid nanoparticle has a very precise and targeted delivery mechanism. So maybe you can work with BSI, find a gene editing therapy or CRISPR therapy, and plug it in with the lipid nanoparticle. So you can send exactly this modified gene or mRNA to a very selective targeted cell that we would like to improve. Because one of the beauty in this mRNA technology is not the mRNA technology itself, but the lipid nanoparticles that I learned. This is the most complicated thing. That you can use this for a very targeted delivery mechanism for drugs, treatment, or whatever it is, for a certain, targeted cell that we would like to cure. So if you can move phase by phase from only, infectious disease vaccine to cancer treatment, and then for gene, some protein replacement, and then goes to the specific delivery mechanism for targeted therapy using the lipid nanoparticle technology, it will be wonderful. So that is the case. Now, again, I love to see implementations. After it was implemented, I persuade them to bring the machine. Now the machine is here in Biopharma. Coba halaman 7, halaman 7. Next. Ini aja. The machine is here. I have the mini one. I asked Biopharma to buy the mini one. The mini is only, you can do research until preclinical phase end-to-end using mRNA technology. You can play around. You want to design in computer in your desk for rabies vaccine, for dengue vaccine, for whatever, chikungunya vaccine is there. And you can come to a prototype. This is the beauty of this machine. If you want to produce, then we can upgrade the machines. Actually, in Ibu Rizka, I don't know whether Biopharma, because we give this for free to Biopharma, the mini. It's our machines. But I said to Biopharma, you have to buy the MIDI. So the MIDI you can produce commercially. So when another pandemic happened in Indonesia, we have the existing infrastructure to produce the vaccine in a very rapid way. 3 months produce. Because designing mRNA vaccine is much simpler than virus-based or protein-based or viral vector-based. Gitu ya. So hopefully we have the mini and MIDI. But the MIDI, the capacity is not big. But at least we can squeeze in, push in the production for a certain vaccine that we believe are very critical if there is a new pandemic. And then if you would like to scale up, we can put several MIDIs and become vaccine. Yeah? So we can have from research and until massive production. But as I said, stop the production immediately, I'm happy enough. And the second thing that I talked to Ibu Rizka is, let's use this mini for research mRNA research platform. So make it open. So the researcher from University of Indonesia, from Bandung Institute of Technology, from Pajajaran, from UGM, from Universitas Airlangga that has been proven producing the vaccine, to use this machine. So they get used to it. So all the smart researchers across the country understand, hey, oh, this is the way if we want to produce mRNA vaccine. Hopefully later on, mRNA cancer treatment, and later on, mRNA, targeted therapies or protein-based replications for rare disease therapy. So the skill, the knowledge, the capacity of Indonesian researchers will be upgraded. So if another diseases or outbreak happens, we can quickly develop because our researchers understand as long as we can get the cell, the pathogen, the live pathogen of those diseases, then we can quickly replicate how we can create an mRNA vaccine that can teach our immune system how to respond if the pathogens come into our body. And the next slide-nya ada apa? Coba next slide, Helena. Okay, the rest is all the presentation that I believe Quantum will share it with you. But the machines is here. So please use it for all the researchers, please use it. For companies, you can help the researcher to use it. I hit exactly the time that I need to move to another meeting, but thank you. All the best, Ibu Rizka, and spend your time, to use the machine. And please help Ibu Rizka to deliver 200 clinical trials of medical device, vaccine, or whatever it is, sponsored with the revenue of 1 trillion rupiah in the next 5 years. Thank you.
Baik, terima kasih banyak Bapak Budi untuk arahannya. Saya mohon tetap berada di atas panggung, Pak. Ya, saya mohon dengan hormat, saya mengundang Ibu Direktur Jenderal Farmasi dan Alat Kesehatan Ibu Lucia Rizka Andalusia, dan dari PT Bio Farma Ibu Yuliana Indriati. Kami persilakan untuk dapat naik ke atas panggung. Dengan demikian Bapak Ibu, hari ini kita akan menyaksikan launching teknologi kuantum. Mohon izin kepada Bapak Budi serta Ibu untuk berkenan nantinya meletakkan telapak tangan kanannya Bapak Ibu pada Lumi Glass Podium setelah saya selesai memberikan aba-aba hitungan. Hadirin yang kami hormati, sesaat lagi kita semua akan menjadi saksi atas launching teknologi kuantum. Bersiap-siap dalam hitungan 3, 2, 1. Hadirin kami hormati, resmi sudah teknologi kuantum diluncurkan. Sekali lagi berikan tepuk tangan yang sangat meriah. Kami persilakan Bapak Ibu untuk melakukan sesi foto bersama. Bapak, foto bersama? Oh, langsung bersama. Oke, baik. Kami persilakan untuk dapat kembali dan seluruh hadirin untuk dapat melakukan sesi foto bersama. Saya mohon untuk berdiri sejenak Bapak Ibu seluruh peserta. Kita akan melakukan sesi foto bersama dengan seluruh peserta di siang hari ini. Oke. Bagaimana? Tim fotografer sudah semua ya? Sudah masuk ya? Yuk, kita mulai hitungan mundur. 3, 2, 1. Sekali lagi boleh? 3, 2, 1. Oke. Baik, terima kasih banyak. Baik, terima kasih banyak Bapak Ibu. Kembali kami persilahkan untuk dapat kembali ke tempat dan duduk kembali. Berikan tepuk tangan sekali lagi Bapak Ibu untuk kita semua tentunya hari ini. Dan kami juga ingin mengucapkan terima kasih kepada Menteri Kesehatan, Bapak Budi Gunadi Sadikin, atas kehadirannya pada acara seminar inovasi mRNA, mengintegrasikan ilmu pengetahuan, regulasi, dan kolaborasi global. Terima kasih. Hadirin yang kami hormati, selanjutnya kita akan melanjutkan ke acara selanjutnya, yaitu diskusi panel. Tentunya pada diskusi panel yang pertama, kita akan membawa tema Strengthening National and Global Readiness for Safe, Equitable and Sustainable mRNA Innovation. Dan sebagai moderator di sesi pertama ini, langsung saja dengan hormat kami mengundang untuk naik ke atas panggung Health Policy Advisor and dari Tony Blair Institute for Global Change in Indonesia. Berikan tepuk tangan yang sangat meriah untuk Dr. Ivan Medika Kurnia. Kami persilakan.
Terima kasih atas perkenalannya. Sebenarnya agak awkward ya karena speakernya banyak yang bahasa Inggris. Jadi izinkan saya melanjutkan dalam bahasa Inggris, Bapak Ibu. First of all, thank you for coming. Allow me to introduce myself in a short time. My name is Ivan Medika Kurnia. Yes, I'm a medical doctor by background, but currently already transitioned to health policy advisor. I'm currently serving at the Tony Blair Institute for Global Change in Indonesia. We have been supporting the Ministry of Health for the past few years. Previously, I supported Minister Budi and Ibu Rizka at the G20 Indonesia presidency, which I think was very interesting because at that time we identified 3 challenges on mRNA manufacturing. The first one is viability, scalability, and then the last one is sustainability. I believe the previous presentations by Ibu Rizka and Pak Budi, we already overcome the first 2 challenges of viability and scalability. And hopefully this panel will enrich our discussions on the third part on sustainability. So without further ado, I will invite the speakers on stage. At this panel, we have 5 speakers joining us mixed in person and online. In person, we have 2 speakers. Professor Shushan, please come on stage. And then the second one, Dr. Ibu Susan, please join me on stage. And then joining us online, we have 3 speakers. Ibu Diah from BPOM, and then we have from WHO, Dr. Mubang Ngizi, and the last one from SEPI. Yeah. Ibu Susan, please come on stage. Ibu Diah Puspitasari, I hope you already joined us online. Dr. Mubang Ngizi, and then the last one, Martina Ox from SEPI. So the discussion will be, the sequence would be the presentation by each speakers for 15 minutes. Hard stop please, 15 minutes. And then followed by discussion. Dr. Susan will present first with the timer on the left one, followed by Ibu Susan, and then we'll move on to our online speakers before we head on to the discussion. Prof. Susan, the stage is yours.
Good afternoon. So first of all, it was very inspiring to hear from Minister Budi about the vision for Indonesia, what exactly has been planned. Oh, thank you. So thank you so much for inviting me for this meeting. This looks like very interesting group in terms of what we want to do. What I wanted to discuss basically is first, maybe in first few slides, I will introduce about the International Vaccine Institute, what we do, what exactly is the focus. And then I wanted to highlight 3 different mRNA vaccine development initiatives that we are working at IVI in collaboration with different partners. And I will end with a new funding mechanism that we are trying to develop with Indonesia, India, and Brazil. And that's what we just discussed with the minister. So just to introduce about the International Vaccine Institute, we are basically an intergovernmental, not-for-profit vaccine research and development organization. Our headquarters is in Seoul, South Korea. But I will talk about in the next slide, we have now multiple offices in different countries. The major part of our mission is to discover, develop, and deliver safe, effective, and affordable vaccines, right? So this is very common to most of the people that are in the room. What we also realized during pandemic that there has been an issue about vaccine equity. The pandemic, there were a lot of first things. For the first time, we developed a vaccine within 256 days, from development all the way to licensure. But then there were a lot of issues in terms of the vaccine distribution. That's why we added vaccine equity, impact, and sustainability to our mission, which is what we are working on. If you see, we are about 300 people based, 80% are based in Korea, but 20% in different parts of the world. Each of them actually works on these 4 areas that we talk about. The discover part, which is the laboratory component of what we do, where not only we develop the antigens, the vaccines, the adjuvants, as well as the assays, but we also work with the partners to develop some of those things and transfer those technologies. The second part is under development, which is going from the translational part from lab to the clinics, which is the clinical development, regulatory, getting all the way to licensure. And as we know very well, licensure is important. But if the vaccines are not used by the communities and the countries, it is still not very useful. So the delivery component of what we do is also very important for us. We work in multiple countries to basically understand in terms of what can be done to increase the uptake of the vaccine. And if you see the data coming from various organizations, it takes about 5 years, or actually 7 years, for a new vaccine to be introduced coming from developing country vaccine manufacturers. And if there is a label change, it takes about 5 years after the label change approval for countries to adopt the new vaccine or new indication. So this is a huge lag period. Not only there is a clear lag in terms of the timeline for research and development, licensure, but there is lag also in terms of acceptance and use of those vaccines, which we generally tend to neglect, and this is part of our mission, and we also work on a lot of this for surveillance activities. And you can see on the bottom half of the slide, various pathogens or vaccines that we are currently working on. As you can imagine, like a company, there are a lot of products that we have in the laboratory part. Few of those, they translate into the clinical part. Few of those goes into the delivery part, and then we have the surveillance network that I mentioned about. So this is the global footprint of the organization. We are still quite small, still quite young, I will say, as compared to Biopharma, which is 135 years, I've been told. So we are based in Seoul, South Korea, which is our headquarters. After the pandemic, we have opened a regional office in Europe, which is in Stockholm, in Sweden. We also have a country office in Vienna, in Austria. And then recently, we also opened our regional office for Africa, which is in Rwanda, in Kigali, as well as we have a country office and a project office, which is in Kenya. In addition to that, we recently collaborated with Hong Kong Jockey Club to open a global health institute focusing on pandemic preparedness, which is what Minister Budi clearly mentioned as the priority for Indonesia. And we are working together with Hong Kong University, Cambridge University, and IVI. As an intergovernmental organization, we are being governed by the countries. Indonesia is firmly a major partner in that, as you can see. We have about 55 countries which have either joined the treaty for establishment of IVI or they are in the process. So 44 of them already have completed, and we have 7 of those countries who are the state funder of IVI. And then I will talk about the pipeline maybe very briefly. So this is what we have, the pipeline, the bacterial pipeline, as you can see. And these are not only the vaccines that we are developing at IVI laboratories. These are also the products that we are working collaboratively with different partners from different parts of the world. And you can see their names mentioned. So this is the pipeline on the bacterial part, the parasitic part, and then we do a lot of capacity building. And Minister Budi is not here, but he did mention about the model from Malaysia. So we have been working very closely with Malaysia for training their staff on the clinical trial capacity building, especially focusing on phase 1 clinical trial. Because one of the gaps which was not mentioned in this presentation is Malaysia has done multiple phase 2 and 3 clinical trial, but they don't have the phase 1 clinical trial mechanism at this point in time, and we are working very closely. The second part is we have worked with multiple countries, Nepal, Rwanda, Mozambique, Madagascar, to increase the clinical trial capacity building. And we are working with a very interesting project with Afrigen, which is an mRNA-based Rift Valley vaccine. And we are basically shadowing them in terms of, so the model is Afrigen gets the funding from CEPI, and Afrigen funds part of the process from IVI, and we are going to provide all the SOPs to them. We are going to work together with them on every step of the process so that they get the training on the job in terms of how to conduct clinical trials, also the assay verification, and then from next time onwards, they will be able to take care of this clinical trial by themselves. So this is part of the capacity building. This slide talks about the viral vaccine pipeline that we have. We still work quite a lot on the COVID vaccine, but there are a few other things which is quite interesting. One of the new technologies that we are quite excited about is the microarray patch. As we have seen, after pandemic, there has been increase in vaccine hesitancy across multiple countries in the world, and it's not coming from one single factor. There are multiple factors contributing to that. However, having needle-free vaccine is going to be a way forward, which will decrease your storage space, which will decrease the cold chain requirement. It will also reduce the adverse effect associated with the needle-based vaccine delivery. So we are working with a couple of manufacturers, one based in Korea, for a microarray patch, which potentially can have multiple antigens or multiple vaccines on the same patch. You put the patch on your skin, probably for 3 minutes. It can be reduced up to 1 minute, and then you just take it out. No rash, no issues, and then the vaccine is getting very quickly absorbed in the skin, and you don't face all these problems. As I mentioned, I want to introduce the 3 major mRNA initiatives that we have. There are multiple different initiatives. I chose these 3 because I think these are more relevant to this particular discussion and the seminar. The first one is what we call the development of a decentralized and accelerated mRNA vaccine production system. The acronym is a bit weird, but that's what we came up with. And then second is the pan-sarbecovirus that we are working very closely with Drew Weissman, which I will talk about. And as I mentioned, the third one is the Rift Valley vaccine, which is a collaboration between Afrigen, IVI, and CEPI. So I think we have seen what mRNA can bring to the table during pandemic. This has been a technology which was quite successful during pandemic. At the time of the pandemic, basically, mRNA technology has a lot of success, but we also need to understand what are the limitations and how we can work on those challenges. So I think most of you are aware, I will not spend a lot of time on the diagram which talks about the process for mRNA vaccine development, but on the right-hand side, you can see some of the challenges that are currently being faced for mRNA vaccine technology widespread use. The first and foremost major is the scale-up issues and the production capacity. I will talk about that. The second, I think Minister Budi also mentioned about this, the raw material supply chain, as well as the LNP manufacturing and IP issues that are there. And the 7 number, which I highlighted here, is the high production cost. If we have to use mRNA as a major technology for vaccine delivery for multiple different vaccines, we need to really work on these gaps that we have. And the major reason for these gaps is most of the mRNA vaccine technology currently they lack scalability because it's a batch process module, and it increases the cost as well as it has impact in terms of the slow delivery of those vaccines. Obviously, we have seen, and as Minister Budi presented, there are 2 different initiatives which are going from the batch production model to more modular structure of the mRNA vaccine discovery. One is led by BioNTech in Rwanda, which is going to be implemented very soon. They are doing the process validation right now. And then second, obviously, as Minister Budi mentioned, is Quantum. And then we have different systems under Quantum, the Infinity program, and then MIDI, and a few other things that Minister mentioned. I think these are good developments from the initial part of the vaccine technology. They still use, although this is a modular structure, they still use batches, and there are limitations in terms of that. And again, I don't need to spend a lot of time on this, but just to showcase that, the batch production, which is the current model that we are using for mRNA vaccine development as well as production, has limitations. The continuous mRNA vaccine production is going to be much better, but it's going to be very complex. It has a lot of advantage in terms of productivity, yield, cost, scalability, and production consistency, but it increases the complexity quite a bit. So what we are trying to do at IVI, working with Korean government and multiple different partners, is to trying to develop an mRNA vaccine production system to basically go with the 100-day mission that we have seen from CEPI as well as from other partners by developing a microfluidic-based, fully integrated modular platform. And then there are some key advantages of that system that we believe will be important for mRNA vaccine production by enabling precise control, by maximizing efficiency, by having flexible production as well as minimal risk of contamination by using single-use system. And this is the video. If it works, it will be great. So this is that, I don't know if there is a sound for this, but maybe I can explore. So this is a container that can be developed, which can be put at the back of a truck. And this has all the system that we need for mRNA vaccine development. So this is the main production area in the container that we see with different modules that are listed here. The IVT module, the LNP module, and then also the ventilation system. The idea will be for this to be in a container so that that container can be shipped anywhere in the world or within the country, depending on the emergency. If there is outbreak, let's say, of Rift Valley fever in Kenya, in remote part of Kenya, and if we don't have the production facility there, if we have this kind of container which can be shipped to that remote part, it can develop 1 million doses within 100 days or even earlier. We are getting some very interesting results from this that we can probably get 1 million doses in less than 10 days, but we need to get this verification and we need to develop the system very clearly as we move forward. But this can be part of the solution when we talk about different emergencies in remote part of the world. So this is the inside view of the container that you have seen. And again, that was the cartoon that you saw. This is actually the miniature model that we have. And we are taking this model. I could not bring it here because it's still quite big size. And then this actually showcases how the actual truck, we are calling it mRNA truck for lack of a better term. And then this will be one of the mechanisms that we can contribute to the global health emergency as well as the pandemic fund. So we are discussing about that. Right now, we are in the step 1 of this particular project where we are developing each of those modules separately. From January next year, the plan will be to bring all these modules into some sort of unified container system. This will take about 2 years. That's the current plan. And at the end of 2 years, by 2028, we are going to evaluate this technology in terms of its utility as well as whether it achieves the purpose that we are talking about. And as I mentioned, there are 4 different partners collaborating with this, and this is funded by Korean government, about $18 million of funding that we have received from them. The second project that I want to talk, if it moves, is the project that we call the BPCO2, which is the Broadly Protective COVID Panservicovirus, which is a collaboration between University of Pennsylvania with Drew Weissman, who got the Nobel Prize for mRNA discovery, and his team with UNC, University of North Carolina, University of California, Davis, as well as Chulalongkorn University in Thailand. The idea behind this is basically to develop multiple monovalent spike mRNA LNPs that we can combine into multivalent spike VLPs, and understanding what are the current variants of COVID, for example, or different pathogens. This can be used for different pathogens, potential future variants. And one of the things that we need to also think about is the spillover from animal or zoonotic transmission to human transmission. So if you can account for all of that and develop an mRNA LNP vaccine which will be providing the broad protection against one particular pathogen family, that's what the target we are trying to achieve now. And then last project that I will quickly talk about is the Rift Valley. As most of you are aware, this is again a spillover from zoonotic transmission to humans. It is happening in the Horn of Africa quite a lot. There are different transmission cycles in different countries, whether it's animal, human, insects. As you can see there, we are working, funded by CEPI with Afrigen, to basically develop different mRNA vaccine candidates. The clinical trial will be sometime in 2027 that we are targeting. Hopefully with that, we can move forward with the phase 2 clinical trial of this vaccine. And then there is a question about sustainability. We have seen a lot in recent years, recent months, that there has been the global health funding that was there. US government was contributing about 21% of all the available global health funding, right? With the new structure, with the new priorities from different parts of US government, the similar kind of funding is not available. Now what we are talking about is, can we have a global South-led initiative where the countries have their own priorities and they decide what exactly needs to be funded? exactly what Minister Budi mentioned a few minutes back, that each country need to decide their own priorities. So what we are trying to create is the bridging research investment in Global Health Technology Fund, which is going to be about $300 million fund for 5 years that we are working with Indonesia, with Brazil, and with India. Potentially many other countries will join into that. And this is a model that has been used in Korea, in Japan quite successfully. We are trying to bring that now to Global South, and this is something that we are discussing very closely with these 3 countries. So hopefully we can launch this fund by middle of next year. So that's my time. Thank you so much.
Thank you, Prof. Sushan, for the insightful update on mRNA. The next speaker is Dr. Susan Nur. The stage is yours.
Thank you. Good morning. Selamat siang ya, Bapak Ibu semuanya. Mohon maaf. I'm sorry, I'm will deliver presentation in bahasa because according to the committee that all of the speakers, all of the attendants come from Indonesia, so it's easier to them to understand what I. Ya, Bapak Ibu semuanya, saya hari ini akan menyampaikan terkait dengan membangun sistem biosecurity. Jadi jauh ya dengan teknik mRNA-nya yang nanti akan diterangkan dulu, ada Prof. Betty, kemudian ada Prof. Santigala. Jadi di sini kita berbicara terkait dengan biosecurity atau keamanan hayati dan juga biosurveillance system untuk mengamankan inovasi dalam hal ini adalah terkait dengan mRNA ya di sini, karena mungkin Bapak Ibu semuanya selama ini sudah mengimplementasikan terkait dengan biosafety atau keselamatan hayati. Lah di sini lebih ditekankan ke arah keamanan hayatinya. Baik, ini ada 4 hal yang akan disampaikan terkait pentingnya biosecurity dan biosurveillance dalam hal memahami tantangan global, karena kemudian yang kedua terkait strategi integrasi untuk kesehatan dan riset serta regulasinya, kemudian pembelajaran dari model internasional yang mungkin bisa salah satunya diaplikasikan di Indonesia, kemudian penguatan ekosistem dan implementasi nasional. Nah, ini yang pertama terkait dengan biosecurity. Kenapa ya, penting sekali kita mempelajari tentang biosecurity atau keamanan hayati terkait dengan teknologi mRNA ini dan juga biosurveillance. Jadi kita tahu sekarang ini bahwa tantangan terkait dengan ancaman biologi semakin kompleks, semakin meningkat di dunia. Terutama salah satunya adalah mRNA itu berpotensi untuk dual use riset yang dapat disalahgunakan. Jadi seperti mata pisau yang salah satu untuk kebaikan tetapi mungkin akan disalahgunakan untuk kejahan bagi orang-orang yang punya kepentingan khusus. Dan juga kita tahu bahwa mRNA teknologi ini juga nantinya juga akan berisiko untuk bioterorisme, karena di sini mRNA hanya menggunakan urutan dari mRNA yang dimana bisa dibolak-balik dan gen itu, sehingga dapat mengakibatkan terjadinya misalkan peningkatan virulensi dari protein yang sudah dihasilkan seperti itu, dan juga mungkin akan ada toksik atau toksin dari patogen yang akan dipakai. Kemudian juga ada ancaman juga selain dari luar di dalam, yaitu adalah pencurian data terkait dengan data kita yang kita punyai, dan juga terkait dengan kekayaan intelektual atau intellectual property, dan juga kita juga berhadapan juga dengan adanya kebocoran hasil penelitian kalau kita tidak membuat informasi security yang cukup baik, dan juga akan terjadi gangguan rantai pasok, karena kita selama ini untuk teknologi mRNA masih membutuhkan pasokan dari luar. Jadi ini adalah salah satu yang mungkin menjadi kendala, bisa jadi kita nanti penelitian akan berhenti kalau ada yang menyalahkan. Kemudian juga adanya pelepasan patogen kalau kita tidak mengimplementasikan biosecurity terhadap pengembangan teknologi mRNA ini, dan juga mungkin adanya ketidakpatuhan terhadap regulasi akan menghambat hasil daripada inovasi mRNA ini. Nah, sekarang kalau kita berbicara tentang biosecurity ataupun biosurveillance, kita harus tahu bahwa kedua istilah ini itu saling melengkapi, seperti halnya biosafety dan biosecurity yang menjadi biorisk ya, saling melengkapi. Di mana kalau biosecurity lebih difokuskan pada pencegahan untuk melindungi material biologisnya, dalam hal ini mungkin mRNA-nya, kemudian fasilitasnya dan datanya agar tidak ada penyalahgunaan atau pelepasan patogen. Sedangkan kalau biosurveillance kita lebih fokus kepada bagaimana mendeteksi, memonitoring dari ancaman-ancaman biologis terhadap aset kita yang sangat berharga, sehingga tidak terjadi kita akan mengetahui secara real time apabila akan terjadi sedikit gangguan terhadap keamanan dan sebagainya. Nah, kedua, biosecurity tidak bisa berjalan sendiri tapi harus terintegrasi. Dengan mengintegrasikan biosecurity dan biosurveillance kita dapat menciptakan suatu sistem pertahanan yang berlapis, multi-layer ya, jadi bisa mencegah ancaman, kemudian mendeteksi dini, jadi early warningnya, kemudian juga menjaga sustainability atau keberlanjutannya dari inovasi dan juga keamanan nasional otomatis yang terakhirnya. Nah, ini pilar biosecurity. Sebetulnya untuk pilar biosecurity atau keamanan hayati itu awalnya ada 5 pilar, kemudian berkembang menjadi 6. Kemudian sekarang berkembang lagi menjadi 8. Jadi yang pertama itu adalah terkait dengan keamanan fisik, keamanan kehandalan dari personelnya, kemudian keamanan terhadap informasinya, kemudian terkait dengan kontrol dari material dan juga akuntabilitasnya, termasuk transport security dan respon emergency keadaan darurat. Nah, yang kedua sekarang ini ada 8 sebetulnya, yaitu terkait dengan manajemen dan juga biosecurity awareness-nya. Jadi seperti itu. Jadi mungkin ke depannya pilar itu akan semakin luas karena makin banyaknya hal baru ya yang akan kita hadapi ke depannya. Dan kita kalau berbicara tentang biosecurity, biosafety, kita selalu memikirkan skenario terburuk, bukan skenario tidak akan terjadi, tapi selalu skenario terburuk ya. The worst skenario yang kita harus bicarakan terkait dengan biosecurity. Nah, ini untuk strategi integrasi bahwa sebetulnya biosecurity itu tidak menghambat untuk inovasi terkait dengan bioteknologi dalam hal ini dengan mRNA, tapi justru dia akan mensupport karena dapat melindungi investasi penelitian kalau kita mengimplementasikan biosecurity secara benar, baik di industri ataupun di riset dan sebagainya. Kemudian juga otomatis kita akan mendapatkan difasilitasi terkait dengan kolaborasinya, terutama dalam hal ini bahwa dengan adanya kolaborasi kita harus berbicara tentang MTA, Material Transfer Agreement, bagaimana kita bisa benefit sharing antara kedua pihak yang tidak saling merugikan tapi saling menguntungkan. Dan juga otomatis kita adanya data-data yang komprehensif ini dapat mendukung untuk decision maker untuk mengambil keputusan dan juga untuk monitoring secara real time terhadap teknologi yang akan masuk ke Indonesia seperti itu. Nah, kemudian terkait dengan sistem kesehatan masyarakat itu merupakan pintu yang utama. Jadi yang di Indonesia itu memang kita tetap harus kalau dalam teknologi yang maju ya untuk sekarang yang modern, mRNA itu merupakan suatu lompatan bioteknologi modern saat ini. Itu memang kita sudah harus berpikir tentang melakukan suatu sistem surveillance nasional. Dalam arti bukan hanya surveillance terhadap penyakit itu sendiri, tapi terhadap data-data epidemiologi kita dan juga deteksi ancaman biologis yang mungkin akan muncul. Ini nyata, karena memang mungkin di Indonesia kita masih berpikir tentang ancaman biologis belum masuk, masih berupa bom, panci seperti itu ya. Jadi belum berpikir tentang lebih jauh lagi. Jadi di sini juga nanti kita perlu adanya kemitraan kesehatan. Jadi kita harus ada protokol pelaporan kejadian mencurigakan. Nah ini di dalam Fing report di sana di dalam COC Code of Conduct ya dari life science untuk industri itu di sana juga salah satu untuk sesuatu yang mencurigakan, isu-isu itu tuh harus dilindungi, orang-orang yang menyampaikan itu juga harus dilindungi. Kemudian juga sistem kesehatan ini juga akan kita berkolaborasi antar lintas sektor termasuk dengan veteriner karena ini banyakkan penyakit-penyakit zoonosis juga yang akan menjadi wabah pada outbreak untuk kesehatan manusia, kemudian kemudian terhadap keamanan nasional serta jejaring internasionalnya. Nah, Bagaimana tentang peran institusi? Dari institusi riset tadi yang disampaikan oleh Pak Menteri, memang dalam BRIN, kebetulan saya dari BRIN, ya itu adalah kita masih dari basic sampai mungkin kalau Purwarupa sudah mencapai TKT 718, tapi mungkin nanti ke depannya sudah harus hilirisasi sampai ke industri, itu juga harus ada penerapan. Nah, institusi laboratorium itu harus mempunyai standarisasi keamanan. Dalam arti penerapan BSL sesuai dengan analisis risiko. Nah, inilah yang jarang dilakukan di Indonesia terkait dengan analisis risiko karena menganggap ini sulit. Tapi sebetulnya dengan analisis risiko kita bisa mencari mitigasi yang tepat yang dapat kita gunakan untuk menanggulangi kemudian manajemen informasi terkait dengan database untuk penelitian dual use. Indonesia punya COC, Code of Conduct Life Science, tetapi memang sosialisasinya belum sampai. Tapi kalau di Singapura itu sudah digunakan dalam industri, jadi ada beberapa hal yang terlindungi. Kemudian pengembangan kapasitas jejaring keahlian nasional global, kemudian pelatihan itu sangat penting sekali terkait dengan biosecurity. Terutama dalam penelitian itu biasanya semua riset yang menggunakan bahan biologi berbahaya harus sudah direview oleh IBC, Institutional Biorisk Management, yang akan memberikan approval boleh lanjut atau tidak dengan melihat kompetensi dari SDM, kemudian melihat ke arah fasilitas yang ada, kemudian terarah dengan biosafety dan biosecurity-nya. Nah, kemudian ini bagaimana kerangka regulasi untuk manufaktur mRNA? Karena mRNA itu berpotensi untuk menjadi dual use, berpotensi untuk menjadi bioweapon, maka kita harus ada otorisasi yang kuat terkait, kita harus melakukan analisis risiko secara menyeluruh, kemudian harus melakukan inspeksi fasilitas di manufaktur yang memproduksi, dan juga kita harus bisa trace back atau tracking dari produk dari riset hingga ke distribusinya itu. Kemudian juga ada pengawasan yang sifatnya sustain. Jadi di sini ada post market surveillance-nya, pelaporan apabila kejadian mencurigakan, juga pemantauan yang berkala. Yang terkait harmonisasi internasional itu juga sangat penting karena kita harus mengadopsi standar WHO otomatis ya, termasuk juga dengan ICH International Council Harmonization ya, terkait dengan kemudian harus ada mutual recognition agreement dengan badan regulasi internasional, termasuk juga partisipasi aktif dalam forum-forum global. Nah, ini dalam biosecurity dalam produksi RNA. Jadi teknologi RNA itu memerlukan perhatian khusus. Kan tadi saya katakan bisa disalahgunakan menjadi dual use dan juga bisa jadi potensi untuk bioweapon. Sehingga kita harus berpikir lebih jauh bagaimana kita melihat risiko spesifik terhadap teknologi mRNA ini. Dalam arti kita harus dapat memberikan keamanan rantai pasokan, kemudian pelindungan terhadap formulasi, itu semua adalah valuable material yang agen ya, yang harus betul-betul kita lindungi karena ini sangat penting untuk intellectual property kita. Kemudian harus ada protokol keamanan manufaktur. Di sini harus ada kontrol fisik dari fasilitas produksi, adanya akses kontrol yang hanya orang-orang tertentu authorized person yang bisa masuk ke dalam daerah manufaktur itu, dan juga adanya reliability atau kehandalan dari personel. Kita harus berpikir bahwa kejahatan itu bukan dari eksternal, dari insider juga. Insider, outsider itu juga sangat berpotensi untuk menimbulkan gangguan. Kemudian juga quality assurance juga kita harus integrasikan untuk tracking batch-nya. Nah, kemudian kalau pembelajaran internasional, kita lihat di model di Amerika Serikat, mereka mempunyai struktur kelembagaan, sistem integrasi. Ada CDC untuk surveillance, ada FBI, ada FDA, dan juga NIH untuk riset. Nah, juga sistemnya terintegrasi dalam BioWatch program di situ. Nah, termasuk juga adanya monitoring laboratory response network di situ. Jadi ini pembelajaran dari Amerika itu adalah pentingnya koordinasi atau antar agensi investasi teknologi dan deteksi yang canggih ini. Kalau di model Uni Eropa juga ada CDD, namanya European Center for Disease Prevention and Control. Ini koordinasi surveillance lintas negara, dia akan melakukan rapid risk assessment ya, kemampuan itu, dan juga adanya TESI atau European Surveillance System berupa tool yang digunakan. Ada juga biosafety dual use research dan juga regulasi harmonisasi. Nah, kalau di Asia sendiri di Singapura itu bisa lebih relevan dengan kondisi di Indonesia. Kita mungkin bisa menjadi salah satu mengadop dalam hal ini, yaitu karena dia ada juga National Public Health Laboratory, di Indonesia juga ada tata kelola biosafety terkait dengan IBC-nya, kemudian ada ekosistem inovasi, adaptable untuk juga Indonesia jadi ininya. Nah, kalau penguatan ekosistem, ini harus kita ada, Indonesia kalau akan melakukan implementasi biosecurity harus ada roadmap implementasi 3 fase, dari mulai fondasi, penguatan institusi, kemudian integrasinya mengimplementasikan dan juga kedepannya operasional penuh. Mungkin Indonesia bisa menjadi pusat inovasi bioteknologi nasional kedepannya. Ini ada beberapa elemen utama di Indonesia, kita ada kelembagaan, ada teknologi, ada regulasi, dan ada SDM. Dan semua ini sudah menjadi satu yang sudah banyak diimplementasikan di Indonesia. Dan ini adalah sinergi biosecurity adalah kuncinya, dan juga dukungan ekosistem sangat penting sekali. Nah, outcome yang diharapkan mungkin ke depannya Indonesia bisa sebagai ASEAN biosecurity leader maupun sebagai industri biofarmasi yang aman dan inovatif. Nah, ini pesan kuncinya, jadi harus ada sistem biosecurity dan biosecurity terkait dengan persyaratan inovasi berkelanjutan untuk teknologi mRNA, ada model internasional yang kita bisa ambil, kemudian Indonesia mempunyai kapasitas juga besar untuk menjadi regulator leader. Ini referensinya. Dari sini kita tutup dengan mari bersama-sama kita membangun sistem yang melindungi inovasi kita sambil memfasilitasi kemajuan ilmu pengetahuan dan kesejahteraan masyarakat. Terima kasih. Thank you very much.
Terima kasih banyak Dr. Susan. Mungkin saya akan lanjut ke speaker kita yang ada di online, online speakers. Pertama mungkin saya akan invite Ibu Diah dari BPOM. So the third speaker is Ibu Diah from the National FDA. Ibu Diah, the time and screen is yours. Thank you.
Okay. Okay, thank you. Good afternoon, everyone. Thank you for the opportunity. And my name is Dia, and I'm the team head of the biological product registration in BPOM. And it is a pleasure to speak with you today. And I'd like to begin with introducing the topic of my presentation is enhancing national regulatory readiness for novel mRNA manufacturing and products. And next. Okay. BPOM as the national regulatory authority in the field of drugs and food Indonesia has a surveillance system that covers entire spectrum, including pre-market as well as the post-market surveillance, law enforcement, and public education. And for pre-market evaluation of drug is carried out based on the evidence of safety, efficacy, and quality, as well as the compliance with manufacturing facility requirement in accordance with the good manufacturing practices, or we call it GMP. And during the research and development stages, BPOM provide guidance before and during non-clinical and clinical trials. For post-market surveillance includes GMP, good distribution, as well as the GDP, good distribution, practice inspection, sampling, and laboratory testing, monitoring of product advertisement and labels, as well as surveillance of product circulation and pharmacovigilance. Post-market surveillance is conducted through sampling and testing of drugs in circulation, as well as inspection at manufacturing, distribution, and healthcare facilities. And BPOM has already implemented for electronic based public services for enhanced service quality and making it more efficient, effective, transparent, and accountable. Next slide. This is our highlight for the new regulation that BPOM is continuously develops regulation that are flexible and adaptive to technology advancement and global trends, including the growing trend of the product that based of the mRNA technology. And the regulation related to drugs are reviewed periodically, and if necessary, new regulations are created, and existing regulation that no longer align with current development will be revised or replaced. And in the slide, some of the BPOM latest regulation includes the BPOM Regulation 14/2024, replacing the previous one for governing online drug and food distribution. And then BPOM Regulation number 7, 2025, revising number 7, 2024 for the guideline of the GMP. And BPOM Regulation 24, 2017 is the criteria and procedure of drug registration, and right now is currently being revised. And we have also several addendums on this regulation. And the last one is the BPOM Regulation number 8, 2025 for the guideline of evaluation of advanced therapy medicinal products. And since BPOM also adopt the international guideline, we also refer to the WHO. And for the product based mRNA technology, WHO having a certain regulation, that WHO TRS, that to evaluate the quality, safety, and efficacy aspect of the messenger RNA, but it is only for the vaccine-based mRNA. Next slide. Okay. For the next slide, in general, drug product development begins at the laboratory scale stage. And laboratory scale development, including in vitro and preclinical testing in animals, must comply with the GLP, good laboratory practice, to determine the pharmacological and toxicological effect of the new drug candidate. And the next stage is the pilot scale and production scale development. Products that show satisfactory in preclinical result may proceed to clinical trials in human subjects. And clinical trials must be conducted in accordance with the GCP, using products manufactured in a facility certified for the GMP. And the clinical trials consist of the 3 phases in the pre-market before approval is phase 1 on the healthy subject, phase 2 on the target population, as well as to determine the dosage. And phase 3 on the large number of the subject to confirm the efficacy and safety profile of the new drug candidate. And next slide. In the downstreaming new drug development, BPOM provides early guidance through the investigational new drug, or we do the drug development assistance. We call it OPB, Obat Pengembangan Baru. This includes consultation regarding development of active substances, manufacturing processes, analytical methods, and non-clinical testing needed to support the submission of the new drug approval. And the submission of the new drug candidates may be done from phase 1, 2, or 3, and consultation can occur before, during, or after the clinical trials. Next slide, please. Okay. For the next slide is about the registration process of the mRNA-based product. That the applicant must submit the registration dossier to our online system application. We call it New IRO. And after the submission of the dossier, we do the evaluation in 3 main aspects. The first is the safety and efficacy, which we can do this evaluation from the non-clinical and clinical data. And then we do the evaluation on quality for the drug substance and drug product, as well as the compliance for the current GMP. And last but not the least is the product information, that the information must be clear, objective, and complete to ensure that the use of the drug is used in the used rationally. And we also do the evaluation of the RMP, risk management plan. And then for recommendation of the new drug application, we involve the national committee as well as the ad hoc clinician expert to assess whether the benefit is greater than the risk or not. If the benefit is greater than the risk, and then the marketing authorization, or we can also issue the EUA, emergency use authorization, might be issued if the benefit greater than the risk. Next slide. Here is the acceleration scheme for the registration in BPOM, that for the way for registration requires up to 300 working days. However, to support the accelerate product availability in Indonesia, BPOM has implemented accelerated pathway. And there are some acceleration scheme. The first is the 50 working days for first time registration of newly developed vaccine by pharmaceutical industries investing in Indonesia. And then the second one is the 90 working days for the new registration or even the variation of the indication or posology approved in at least one country with a well recognized or established evaluation system. We recognize 7 countries or 7 regulatory authority that we refer. The first is US FDA, EMA, TGA Australia, PMDA, Health Canada, MHRA UK, and Switzerland. And the last one is the 100 working days. This is for the National Health Program. So the product that included in the National Health Program can be accelerated in 100 working days. And the next is for another 100 working days is for first vaccine registration by pharmaceutical industry investing in Indonesia, as well as that the product that have undergone the new drug development or IND or OPB, obat pengembangan baru, and completed at least one clinical trial in Indonesia. Next slide, please. Okay. Good manufacturing practice is the fundamental basis throughout the life cycle of mRNA-based product, from development to manufacturing and marketing authorization. And due to the complexity and sensitivity of mRNA technology, every process must meet a strict regulatory standard. And the C in the CGMP stands for current, so which emphasize that the manufacturers must follow the latest system, processes, and technologies to meet required quality standard. This is essential to ensure mRNA products remain safe, effective, and consistent. And in Indonesia, GMP for biological products, including mRNA, is based on the GMP Regulation Number 7, 2024, and its amendment Number 7, 2025. And for biological products, including the mRNA product, Annex 1 and Annex 2 are mandatory as they regulate sterile manufacturing and biological process control. Okay, next slide. Okay, this is the manufacturing aspect of the mRNA-based products, that the manufacturing process differs significantly from the conventional pharmaceuticals. All mRNA-based products are regulated as the biological products due to their component derived from the biological systems. And this includes mRNA vaccines, and might be the gene therapy that contain the mRNA. And the manufacturing requirement include some control in the manufacturing aspect. This is include adequate control of the material, including the DNA template, enzyme, nucleotides, lipids, etc. And then adequate control of the manufacturing process from in vitro transcription to lipid nanoparticle LNP encapsulation. Since we already know that for the mRNA delivery system is most of the product is encapsulated by the lipid nanoparticle. And then strong emphasis on the process control strategies, comprehensive characterization of mRNA and LNP structure, purity, biological activity, and physical properties. And then release testing for drug substance and drug product, stability testing to ensure product safety and quality during storage. Next slide. Okay, this is the unique characteristic of the platform technology that in the mRNA-based product that as we see, this is the mRNA. That mRNA is produced enzymatically, as we know, through the in vitro transcription. And mRNA includes coding region, untranslated region, 5-accent cap, and 3-accent poly-A tail. And this unique characteristic make the mRNA-based product have the certain complexities. So these are the certain complexities of the mRNA-based product, and I will highlight for the high variation of the purification of mRNA, and then the encapsulation process with the LNP, etc. And as well as in the distribution that mRNA-based product need ultra cold chain. Next. Collaboration between three elements, the academia, business, and government is really crucial to advancing research development and improving access to safe, effective, and high-quality medical products. And Indonesia has taken strategic steps to strengthen collaboration and leverage academic expertise. Academia Business Government ABG model plays an essential role in drug development and regulation. And for role of academia is conducting the applied research, academic oversight, expertise, research funding, and research. Provide scientific foundation through technical trainings, and role of business implement business communication, regulatory support, technical training, and ensure develop product meet regulatory and quality standard.
Yeah. May I ask you to give your concluding remarks because it's already past 15 minutes. I'm sorry.
Oh, okay.
Thank you.
Oke, ya, tinggal sedikit lagi sih Pak. Okay. And then role of BPOM is oversee drug development through regulation, technical training, and research center, provide legal framework and guideline to ensure compliance with safety, efficacy, and quality standard. Next slide. Okay, this is, we also endorse research and the development of the local product in Indonesia. Next. Okay, this is the key takeaways of the ending of the slide, that the mRNA-based product have the unique characteristic, and the LNP technology for the delivery system of this product is really important and is a critical aspect to be considered. And also the control of the raw material and also the manufacturing process is really critical during the manufacturing of the mRNA based product. And I think we also need collaboration with the international agency or international pharmaceutical industry for innovation in the future to support the local production. And we also endorse for enhance the capacity, continuing capacity and regulatory excellence. Okay, I think the end of my slide. Thank you.
Thank you, Ibu Dia, for your Thank you, Ibu Dia, for the insightful presentation, especially from the views of Indonesian FDA. I was also informed that you will leave the room by 3 p.m. For another agenda. So in case any participants would like to ask questions to Ibu Dia or BPOM in general, please relay the questions to me or the committees, and we can ask the BPOM to answer it later. I'll move on to the fourth speaker, Dr. Mubangizi. I hope I didn't mispronounce your name. Sir, the stage is yours.
Yeah, thank you so much. May I be given permission to share my slide? Because, okay, now I think I can now. Okay.
Are you clear now?
Do you see my slides?
Not yet. Oh yeah, now we can see it. Yeah, you can proceed.
You can see it?
Yeah.
Okay, thank you so much. So first of all, to thank Indonesia for inviting WHO to present in this important workshop, and also to deliver my greetings from the Assistant Director General of WHO, Dr. Yukiko Nakatani, who was not able to be with us here. And also to thank the previous speaker from Barimpo, who has ably covered, I think, the concepts of assessment of mRNA vaccines. So what I've decided actually to focus on in the little time now I have is to tell you more about the mRNA technology transfer program, for which even one of the manufacturers from Indonesia participates, but also the aims on facilitating geographically diversified capacity for manufacturing of health products, particularly mRNA-based vaccines. So this program was created with a clear mission to shift technology from being concentrated in a few countries to a globally distributed capacity. We saw that when manufacturing is concentrated in a few geographies, that could be interrupted either by lockdown, as we saw in COVID, or disasters like earthquakes or typhoons and all that, and the nuclear disasters, and this may affect access to health products. So for global health security, it is important that we have geographically diversified manufacturing, especially of important technologies like these ones that we have heard from the previous speakers that are critical in the global capacity to respond to emergencies. So it is about improving health security in low and medium income countries by enabling sustainable regional production of mRNA vaccines and other health products. And this program is about building human capital, the scientists that we need, the engineers that we need, the regulators that we need, that countries need to adopt, regulate, and innovate with this platform. The work we present today shows how this mission has moved from the vision, just being a vision, to actual implementation. And this is remarkable in just a period of 4 years. So when we look at this, the core purpose of the program is simple. It is to ensure that low and medium income countries are not left behind in future pandemics because of limited manufacturing capacity. That's the fundamental. This means establishing and strengthening facilities, transferring the know-how to run them, and enabling countries to produce high-quality mRNA products. Building regulatory and biomanufacturing expertise is equally essential. Without the workforce and NRA capability, manufacturing investments do not translate into access. Ultimately, our mission contributes to global health security through sustainable, reliable manufacturing ecosystems. Unlike bilateral licensing or one-to-one partnerships, the program uses a multilateral model. This allows multiple manufacturers to benefit simultaneously from the same platform and from each other's learning. Regional research and development consortia help shape the pipeline and ensure that products respond to real epidemiological needs rather than global market priorities alone. And the management and oversight are essential, and therefore WHO and the Medicines Patent Pool and partners ensure that standards, accountability, and alignment are ensured across the network. So looking at the program is built around three interconnected pillars. One is the South African consortium that is composed of Afrigen, BioVac, and partners that develops the platform and provides training and hands-on technology transfer. Then there are 15 manufacturing partners across low and medium income countries that are receiving and adapting the platform for local and regional needs. It's important to say that adapting this for local and important needs, and we've heard from the previous speakers how this is translating in the different, even in the local setting in Indonesia. Then we have R&D consortia that build together researchers and manufacturers to develop products relevant to regional disease settings. And again, you know that research is very expensive and complex, but bringing the capacities of different institutions and academia, research institution and academia together, that can enhance the capacity, especially in low and medium income countries, and also help adapt and adopt to relevant regional disease burdens. This model is intentionally multi-layered to ensure technology transfer is not only transactional, but also transformational, stimulating innovation and product development in low and medium income countries. A lot has been achieved. Just in 4 years, the program has created a functioning global network of 16 manufacturers in low and medium income countries. The South African consortium has successfully developed an mRNA platform, transferred the know-how, and trained hundreds of scientists, engineers, and regulators. Facilities in several regions are now progressing towards good manufacturing practice readiness, analytical method transfer, and scale up. Importantly, partners are beginning to apply to the platform to additional diseases, showing that the model can extend far beyond COVID-19. These achievements confirm that low and medium income countries can learn, adopt, and innovate with advanced platforms when the right support ecosystem is in place. COVID-19 exposed structural inequities in access to advanced vaccine technologies. In low medium income countries, we know that waited months, sometimes even more than a year, for mRNA vaccines. The program was created to avoid repeating this scenario in the next pandemic. So phase 1 focused on creating the foundation, that is platform development, training, and initial technology transfer. We are now moving to phase 2, which shifts towards real world impact. And in this product pipeline development, sustainability planning, as we also heard from our colleague from IVI, regulatory and good manufacturing practice, grade manufacturing. That is going to be the focus of phase 2. It also aligns with the WHO pandemic agreement, which reinforces the need for distributed, sustainable, and high-quality production capacity. The program strategic vision for phase 2 is to move from we can manufacture to we can deliver. This includes 3 dimensions. One is commercial viable products developed through research and development consortia and in-license. 2, sustainable operations supported by business plans, market analysis, and financing. And 3, integration with health systems, ensuring products can move through clinical evaluation, regulatory pathways, and into national programs. A key lesson from the first phase is that technology transfer alone is not enough. Countries must also have demand strategies, regulatory readiness, and long-term sustainability plans. Indonesia has been part of this journey, and let's have a few reflections on biopharma that has participated in this. Indonesia's biopharma is a strong example of this vision in action. It has moved from platform installation to planning GMP operations, developing mRNA prototypes, and aligning with the national health priorities. Their work shows how a program partner can move beyond manufacturing into pipeline development and health system integration, which is exactly the ambition of the phase 2. Phase 2 is built around 3 strategic pillars. 1, product technology development driven by research and development consortia, co-development agreements, and in-licensing strategies. 2, regulatory strengthening and convergence, enabling faster, predictable pathways to evaluate mRNA products across regions. And 3, market access and demand forecasting, ensuring that products generated by the program have a clear route into national and regional procurement. These pillars are supported by key enablers like legal and IP guidance, policy support, manufacturing scale-up, and engagement with governments and regional blocs. And again, the example of biopharma's participation directly advances all the 3 pillars through contribution to research and development discussions, progress towards regulatory preparation with BPOM, as we've just heard, and involvement in shaping demand strategies in Southeast Asia. Indonesia's example demonstrates how national leadership combined with global support can accelerate transition from training to real-world impact. The program long-term goal is clear. That is a self-sustaining mRNA ecosystem in low and medium income countries by 2030. On this path, early development business planning, IP strategies, and expert group help de-risk investment. Late-stage development, clinical evaluation, and regulatory pathways bring products towards market entry. And revenue generation and diversified product portfolios then sustain facilities and enable continuous innovation. This journey requires aligned investments from governments, the private sector, and the development partners across the full research and development to delivery spectrum. The mRNA program is more than a technology transfer project. It is a global public good that demonstrates low and medium income countries' capacity to build world-class biotechnology capabilities. Phase 2 is the moment where potential translates into tangible products, regulatory readiness, and pandemic preparedness capabilities. Partnerships remain in the engine of the program across continents, sectors, and institutions. By investing now in research and development, regulatory convergence, and manufacturing ecosystems, we ensure faster, fairer access to critical technologies during future emergencies. WHO remains fully committed to supporting partners, including Indonesia and Biopharma, in building a resilient, sustainable, and equitable mRNA ecosystem capable of responding to future threats. I thank you so much.
Thank you, Dr. Mobangizi, especially the closing reflection. That's really beautiful to hear. Moving on to the last but not least speaker, Dr. Martina Os from CEPI. The stage is yours.
Thank you very much, and thank you very much to the organizers to have CEPI here presenting. And before I start, maybe just, I think you're all familiar with CEPI more or less. Just one very short introduction. CEPI was founded coming out of the Ebola epidemic in West Africa, and it was in 2015, where the response to the health emergency fell quite short, and it was recognized that an organization such as CEPI would be needed to address that something like that doesn't occur at the same in the same scale in the future. So the mission of CEPI is to accelerate the development of vaccines and other biologic countermeasures against epidemic and pandemic threats so that we can be accessible to all people in need. And with that, I go to the next slide. Can you show the slide mode so that the people can see the slides?
It's moving from our end. So.
Yes, perfect. So you may be familiar with this slide. I think it's animated. Can you click through to the end of this slide? Thank you. So I think you're familiar with it, but I find it every time when I see it impressive again, because it really highlights the necessity for reacting very fast to any health emergency. And it as well demonstrates how quickly during COVID the first vaccines were ready. And you all know that the very first vaccines were mRNA-based vaccines. So it really shows that the earlier you can intervene, the more success your intervention has and the fewer cases of any disease you will have. And in the next slide, coming out of that, CEPI developed the 100 days mission, and the mission is really vaccines should be ready for initial authorization and manufacturing at scale within 100 days of recognition of a pandemic pathogen when appropriate. And in order to achieve that, as you all know from your own experiences in vaccine development or biologics development, in order to achieve something like that, you have to be super well prepared. You really have to shift your efforts into preparedness of scientific toolkits, the development infrastructure, the policies, all of this. And that requires a paradigm shift, and that requires a lot of investment up front. Going to the next slide, please. So platforms are a critical part of that. And in CEPI, we use the term platform technology as these are systems that use the same basic components as a backbone and can be adapted to use to against different pathogens by inserting simply new genetic or protein sequences. And then these techniques will be ready set up for rapid use against any disease X. You know that CEPI calls disease X the unknown future pathogen that may break out at some point. So, and as you know, RNA, we heard this from our previous speakers, RNA is particularly suited for this because it is a cell-free technology. It is a very fast to develop technology. It has the platform potential, meaning you can use a lot of data that were generated with one pathogen and apply them to any future, maybe different pathogens, simply because you are limited to switching the antigenic sequence. And we have seen that as well again during COVID, how quickly the technology was adaptable to different variants. Going to the next slide. So how we look at platforms at CEPI is just a very quick overview. What you see here are kind of fictive platforms. So we look at different aspects. We look at the adaptability. We look at the suitability for a certain antigen. We look at the regulatory aspects. We look at the compatibility, which means is it compatible with a given disease. And we look at the manufacturing readiness of our developers, and we look as well at the regulatory readiness. And going to the next slide. This is just a reminder of what has been achieved with COVID-19 vaccines. So a number of companies have achieved late-stage clinical development or received an authorization from a WHO-listed authority. They are the usual suspects like Moderna and BioNTech, Pfizer, but you can see as well that there are some others that have more regional authorizations. Going to the next slide. It captures the landscape of the pipeline against viral targets. So you can see that there is a large amount of activity in the preclinical space, whether that's on RNA modalities or on the delivery vehicles for the RNA. So what you can see in the color coding are for the RNA different modalities. Red is circular RNA. This kind of teal colored is the modified linear mRNA. Then the greenish is the non-modified linear RNA. The brownish is self-amplifying RNA, and the purple is circular RNA. So you can see how diverse the research field really is in different RNA modalities. And it's maybe less diversified for the delivery vehicles, where you see in orange the LNP based and in the grayish, dark grayish, the non-LNP based. But you can see as well in the light gray colored that whether it's the RNA or the delivery vehicle, that there's a lot of research going on where the developers or the researchers have not disclosed really exactly what they're doing, and that is not surprising, as you know. There is limited activities in the clinical space, not surprising with such a new technology, and as well, of course, mainly in the early clinical phases. And I just remind you that this is not the pipeline for RNA products in general, but that just the pipeline against viral targets. Going to the next slide. I highlight now where CEPI is investing. You know that CEPI is funding different aspects of vaccine development research, and we are strategically investing in different areas of RNA-based vaccines technologies. So we are investing in RNA modalities and delivery innovations. We are as well investing in what we call enabling technologies. These are more technologies that facilitate the development and the manufacture of RNA-based biologics and vaccines, but that are platform agnostic, such as DNA, synthetic DNA mainly, or maybe analytical methods, or maybe raw materials. We are as well investing very concretely in platform development, where we are looking at where we are using platforms for our priority pathogens or prototypic pathogens that are well characterized to really develop the platform components with specific developers that we are funding, and biopharma is actually one of them. And we are as well investing in manufacturing innovations, and we are investing in manufacturing facilities network. Against biopharma is a partner in this facilities network. Going to the next slide. It's going a little bit more into detail, giving you just a little bit more insight into where we are investing in terms of RNA modalities. So we have the majority of our projects is still on linear modified mRNA, but we have as well a significant number of projects that are looking at self-amplifying RNA. We work as well on trans-amplifying RNA, and we have few projects that use circular RNA. Of course, as you can imagine, some of those are in very early preclinical stages. You really could call it more discovery, but that is as well where CEPI has a lot of investments in general in the innovation space. Going to the next slide, it highlights the delivery vehicles. Shows that we are investing in several next generation LNP projects. We are investing in alternative delivery methods, so non-LNP based methods, but we have quite a number that use the more traditional LNPs. The next slide is highlighting more the manufacturing innovations, and here we are looking now at the 100 days. Really, the 100 days is very much focused on speed. Get as quickly as possible to a prototype candidate against the unknown disease X that how do I get this out of my screen? Sorry. That focuses on self-remanufacturing, the platform optimization, and the analytical message to get very quickly to the clinic. While the next 100 days are really focusing on scaling up at access. Scale means make it at enough quantities available, and access has several aspects. It really goes into the use in the regions where it may be needed, such as for this you need to look at thermostability, but as well the cost of goods and the ease of administration. And the next slide is looking at some of those manufacturing investments. I think the previous speakers already highlighted the difference between the batch-based manufacturing and more continuous manufacturing. So we have several projects that are in early stages of development that look at a continuous manufacturing or non-batch-based manufacturing, and some of those investments are co-financed or jointly funded with Welcome Leap. Welcome Leap had launched several years ago a program they call R3. R3 stands for RNA Readiness and Response. And we have as well a couple of others. So a diversity of those type of technologies where we are investing. And in the next slide is summarized. Can you go to the next slide, please? Is a little bit more about the enabling technology. So we are investing into DNA template, synthetic DNA template, where we have project with DNA script, and we have as well a project with Afrigen. Both of those are on synthetic DNA, which would be much faster manufactured than plasmid-based DNA templates. And the next slide is looking at our manufacturing network as to how geographically the facilities we are working with are positioned. The red dots are the ones that utilize RNA, and you can see on the very right biopharma located in Indonesia. And the next slide is just highlighting the calls for proposals that are currently open. So if you are interested as well, CEPI is continuing to invest. We have an open call for proposal about to prepare for future epidemics and pandemics. Where there is innovative vaccine platforms is open for applications as well as new vaccine candidates against CP priorities, CP priority pathways. I think at this point in time only Lassa and disease X candidates are open to receive applications. And the next slide is highlighting another call for proposal that is currently open. It's on analytical technologies that are again, this call is actually not limited to RNA technology, but RNA technology included, and those are the more platform agnostic technologies that are not limited to one specific RNA technology. And with that, actually, I went through my slide deck, and I think I can close here. Thank you. Thank you for your attention.
Thank you, Dr. Martina. I'd like to kindly ask you to stay in the Zoom as we head into the discussion. Dr. Mubangazi, are you still there? I hope you are. Okay, perhaps first question
I'm here, but I will request if there are any questions to me, they better need to look for another scheduled appointment.
I see. Perhaps from the audience, do you have any question for Dr. Mubangizi? Yeah, the lady in the middle. Please introduce yourself and your affiliation and what is your question?
Okay, thank you very much. I'm Erna from ITB, Bandung. So I would like to have questions about, based on our experience until now, so to develop the vaccines. The mRNA vaccines is one of the most expensive to develop. Start with a laboratory scale. So maybe WHO has a share a lesson learned how to overcome this problem. Thank you.
Over to you, Dr. Mubangizi.
Yeah, thank you so much. Very, very good question. And indeed, through the research and development of mRNA, but not only mRNA, research and development generally is expensive venture. But as I've highlighted in my presentation, the approach we have taken is to have R&D consortia. That means getting partners who can contribute from different angles, including researchers, but also funders, to be able to develop products and platforms, and these become a public good, not only owned and they can be transferred to developers and manufacturers to transform into commercial products as public goods. We hope that approach is what will make access to such technology and research and development affordable. Thank you.
Thank you. Is anyone else have a question for Dr. Mubangizi before he leaves the room? If not, I'll move on. Perhaps to Prof. Shushan first.
Thank you.
So you expounded a lot on the mRNA platform and how to tech transfer to other countries from IVI standpoint. But I'm curious from the business standpoint, from your perspective, what is the minimum viable operations for the mRNA facility to stay afloat in peacetime instead of in the pandemic? Because in pandemic, we know there's a demand surge, but how do we ensure that it's sustainable during, let's say, stable demand? Thank you.
So maybe this is a complex question, right? Because this is connected to market, this is connected to business, this is connected to finances, and obviously there are multiple other factors that we need to take into account. Let's take a look at, so in general, we know that vaccine business is complicated. I think right now, if you divide the market, there is obviously the private market, even in the developing countries, and the developed country market, the high income market, which is where we see the PCV 13, PCV, then kind of getting into the 10 billion kind of market zone. But the most of the vaccines that are being developed, especially by the developing country vaccine manufacturers, they are targeting the global public market, right? Which is a very volume-based market, and it's a very price-sensitive market also. So vaccine business, it has its own intricate limitations or problems that we need to think about. Now, when we bring the mRNA technology to that, right? Right now, as we have seen from CEPI, there are multiple products which are licensed. There are multiple products which are in development, phase 3 and beyond. And also, the WHO colleague, I don't think he's online anymore, but really about whether governments will consider mRNA as a public good, and whether this will be coming from the grants or support from the government. At some point in time, obviously, the market forces will kick in, and there has to be a very clear strategy for that. Will there be, and right now we are looking, there are a lot of regional initiatives in terms of getting prioritization for the regional manufacturers, whether it's the PAHO revolving fund. Now in Africa, they are planning for the African, I don't know what is the procurement mechanism. If something has to be done for sustainability from pandemic point of view, whether the mRNA is going to replace the existing technologies that we have for the routine vaccines, that's one thing. Second, if mRNAs are going to be considered as the tools for any pandemic preparedness in the future, how do governments see that part? And in that situation, the market forces doesn't really come into picture. But if it is not the case, then I think mRNA technology has to compete with other technologies, and we need to really work on the challenges that I showed in the slide, as well as highlighted by my colleague from CEPI.
Thank you, Professor. Audiences, do you have any questions to all the speakers here, not only Prof. Susan, but also Dr. Susan, and also from CEPI, Dr. Martina? Any question? If not, I'll ask again. So, Dr. Susan, you mentioned about the mRNA platform and how the biosecurity framework need to adjust to the technological advancement. Do you think our current framework in Indonesia is already enough to adjust or to adapt to the technological advancement? Or do you think, is there any specific changes that you'd like to see perhaps in the next year or so to really bolster our framework?
Okay, thank you. Can I answer in Indonesia? Ya, oke. Terima kasih. Kalau kita bicara tentang framework di Indonesia terkait dengan teknologi ini, sebetulnya Indonesia itu sudah banyak regulasi-regulasi yang bisa kita pakai yang membatasi penyalahgunaan pemakaian bahan biologis, termasuk mungkin adanya biosecurity terkait untuk menghindari penyalahgunaan juga. Namun mungkin regulasi ini perlu kayak ada task force-nya ya. Jadi artinya kadang-kadang memang seperti beberapa industri ataupun laboratorium, sebetulnya kita sudah tahu bahwa harus mengimplementasikan baik terkait biosafety, biosecurity, terutama kalau kita talking about the material valuable agent. Nah, mRNA ini sangat unik. Jadi beda dengan microorganism lainnya yang butuh cultivate untuk memperbanyak ya. Tapi untuk mRNA ini kan artinya kita bermain-main dengan gen ya, yang untuk menghasilkan protein tertentu yang kita inginkan. Nah, artinya di sini kan artinya biosecurity itu ketat tuh karena mungkin kita melihat ke arah nanti penyalahgunaan dual use research-nya, atau mungkin ada potensi untuk biological weapon, atau mungkin ya karena manufaktur itu perlu sekali security yang ketat, bukan hanya terhadap material itu sendiri, tapi juga terkait dengan informasi, transformasi, kemudian juga personalnya. Nah, kalau menurut saya sih sebetulnya mungkin belum tersentuh sampai ke arah mRNA ya. Jadi selama ini kita baru di Indonesia peraturan terkait dengan agen biologi berbahaya, jadi infectious pathogen, dan juga beberapa, kebetulan di Indonesia sendiri kita kan belum ada security sensitivity biological agent yang baru akan sudah ada, kita sudah ada listnya, tetapi memang belum ada yang pengampunya. Nah, mungkin dengan adanya SSBA ini nanti mungkin bisa mengarah penggunaan juga sintetik biologi ini untuk membuat regulasi yang lebih ketat. Artinya kita tidak boleh langsung menganggap bahwa produksi kita itu aman-aman saja. Yang saya katakan itu aja. Terima kasih.
So as we innovate, we need to ensure that the security framework also in place, including in the context of collaboration with other partners and multi-countries as well. Thank you for that, Dr. Susan. Once again, the audience, is there any question? You can ask anything to these esteemed speakers. Not yet? Okay. Dr. Martina, I have a simple question. You said in 2022 there's a call for proposal on thermostability. I think ultra cold chain is really the single biggest blocker for vaccine equity in mRNA, especially in Indonesia's context, the archipelago. Are we going to see the next generation of mRNA that's stable at the standard cold temperature, or is it still far away? Thank you.
So when you refer to that specific call for proposal, interestingly, most of the applications that came out of this call were actually about using microneedle patches to overcome thermostability changes. So that is certainly, as well as was mentioned by one of the earlier speakers, an option to address thermostability. We have in CEPI's portfolio as well a couple of technologies that overcome thermostability through other means, either through the composition of the delivery vehicle or as well, lyophilization, I think, is an option that as well many developers have pursued in the past. So I think there are ways to overcome the challenges of thermostability. But another challenge that comes with those is that they're not necessarily cost neutral. So if you have to lyophilize or if you have to put something on microneedle patches, you have to as well consider whether that is as cost effective as if you have a fully liquid. I'm not saying one or the other, whether it's more or less expensive, but it's certainly something that you will have to consider as well.
Thank you. One last call from the audience. Do you have any questions? Let me count to 3. 1, 2. Okay, we have. So I need to count again the next time. Perhaps the lady from the right first with the gray suit, and then I'll come back to you, Boo.
Okay, hi. So I just would like to have a few questions asking about, because now from the list of the participants, right? So we see about the institutes and research, and along the discussion now coming to upscaling mRNA in future, what kind of challenge that now for the Indonesia as a research now have a challenge to upscaling to the industry like mRNA in future?
Okay. Would you like does your question point to Dr. Susan?
Yeah.
Yeah. Okay. Yeah. Thank you.
Okay, thank you. Yeah, maybe for the upscaling for the mRNA vaccine in the future, yeah, like in Indonesia, maybe we have still have many gaps, yeah, for the scaling up because in Indonesia also actually like BPOM already also explain how to mix the good vaccine with CPOP ya, cara pembuatan obat yang baik ya. For the challenge maybe like the awareness of the human research itself, because as this material it look like is like we see something not cannot be seen directly. So maybe the culture responsibility should be improved because usually like we do with the microorganism, we know about the impact of the microbes, but it's different with the mRNA. But because it looks like it's not directly to causing the disease, but maybe we should be aware about the biosafety for the people who work with the material itself, and then for the environment, we have to know also for the people surrounding the manufacturer, we have to thinking about that. And then about the biosecurity itself, like the biopharma, I know because I have already been there, they already have a good, very, very good biosecurity practices for the implementation. So I mean like the laboratory itself, because in Indonesia, maybe this research not only to be like produced by manufacturer, but some of the academicians also doing research with mRNA. We have to consider that we have to do risk assessment to do the research. I mean, for the institution should be have like institution biosafety committee to review all of the proposal that using that like a material like mRNA. So if the human resources is not good enough for the competence, and then for the awareness for the biosafety or biosecurity, and also the facilita is not fulfill the requirement that should be designed for safety, I think we have to consider maybe we can do like a biosafety measurement, whether we can directly do the research using mitigation, or maybe we can eliminate for that doing research, because we still have many gaps actually. Yeah, so for the scaling up, also like the culture of the people itself, usually for the receive like a messenger mRNA vaccine, they're still thinking, is it safe or not? For us, it's different. We already got a vaccination from when we were child or when we were born, but usually using like a microbe that are like a guilt vaccine or maybe live attenuated vaccine, but this is the different one. We have to give like socialization for them that this vaccine actually is really advanced biotechnology, modern vaccine that can be produced with high scale if we compare using the micros. This should be can be socialization for the vaccine itself for using mRNA. Thank you very much.
Nutrition, you want to add something? No, thanks. Okay. Again to Ibu Nidemin. Yeah.
Thank you very much for the opportunity. Maybe just a quick question for the IV. So we would like to ask actually about the priority for development vaccine in Africa. So is there any lesson in Africa? So is there any lesson learned that we can implement from the development in vaccine in Africa to Southeast Asia, for example, Indonesia? So that's the first question. And the second question for Dr. Martina from CEPI is what is the challenging actually for the development of trans-amplifying and self-amplifying mRNA comparing to conventional one? Thank you.
I'll let Prof. Yushan or Dr. Martina, would you like to answer that first?
The challenges for self-amplifying and trans-amplifying RNA. So self-amplifying RNA, there are some obvious CMC challenges. The molecule is quite long. It's usually over 8 kilobases long, and that brings with it some challenges in the CMC. It is not, of course, those challenges can be overcome, as some manufacturers have demonstrated with their COVID vaccines, but it's a little bit more complicated to make than the normal mRNA, and it may need as well a slightly different formulation in order to be most effective. For the trans-amplifying RNA, so for one you need two molecules. One contains the replicon, the other one has your gene of interest. So you have two linear mRNAs that you need to produce. And you need to fine-tune the ratio between those two molecules. And that can be as well needing a little bit a careful investigation. So I hope that answers the question.
Thank you, Dr. Martina. I'll move on to Prof. Sushan.
No, I wanted to go last because this is probably be a longer answer. This will because you ask a question which is quite complex in many things, right? What we need to understand. Let's talk about Africa first, right? And then I was quite happy to see the slide from BPOM. She is not here actually, but when she presented about various initiatives, she actually put ABC, which is academic partners, no, ABG, whatever that sequence was. B was the companies or business, and G was government funding, right? The good point about Indonesia is you have all that ecosystem within the country, right? What needs to be done? What are the gaps? We can talk more about that. But going to Africa, before COVID or even now, there are multiple gaps in all these sectors. One is the funding issue. So there are a lot of funding committed to Africa, but there is one thing about committing the funding. The second thing about absorption of the partners on the ground in Africa to basically get that funding and meaningfully make the impact, right? Which is lacking right now. There are few initiatives, but not quite a lot. The second part is about business. So right now, actually, IVI, we have transferred a technology to one of the manufacturers in South Africa for oral cholera vaccine. That will be the first end-to-end development of any vaccine technology on the continent. So you can see, there are a lot of gaps on the ground there, right? So there are multiple companies, there are multiple manufacturing initiatives. Few of those are more advanced as compared to others, but this is a long way to go. The first part also is academic partner. Getting the R&D on the continent is a challenge, right? Having fill-finish arrangement to be done, that's a simple part, that's a first step, and that's generally what most of the companies and countries they do. Getting the R&D and developing something from scratch takes years and decades, right? I think that's where Indonesia probably is lacking at this point in time, and that can be done, work together from academia, business with the government funding. The good point is, again, there seems to be a lot of government interest. Having health minister being present here, inaugurate this meeting, laying the vision and challenging the researchers to basically develop the ecosystem with 200 sponsored clinical trials, getting the quantum machine and inviting all of you to basically contribute and use that is a great thing. We don't have the similar kind of thing in Africa, right? So these are very different things that we are seeing in Indonesia versus in Africa. One other point that I want to add is, as these countries in Asia, they are becoming more economically progressive. Obviously, they don't fall under the Gavi eligibility for getting the funding from Gavi, right? But again, many of these countries in Asia, they are not yet high income. So one of the gaps that I see specifically for the routine vaccines is how do we procure from Southeast Asia, whether it's going to be a Southeast Asian mechanism, it's going to be an ASEAN mechanism, or more wider Asian kind of collaboration. How do these countries pull their resources together, negotiate for the pricing with the manufacturers, and develop something as a pool procurement mechanism? Because there are a lot of gaps in terms of the GDP per capita for many of these countries. Maybe that's something also can be thought about in addition to the R&D gaps. Sorry, that was a bit long answer, but...
Thank you. As we're approaching the end of discussion, I'd like to call for the last two questions, perhaps, if there is still any question. No? Okay. Let me quickly recap what we have discussed in the first session, right? From Prof. Sushan, Dr. Susan, Dr. Martina, and Ibu Dia from BPOM, and Dr. Mobagizi from WHO. The first lesson that I think is important, that vaccine equity issue is really being heightened because of the pandemic. And now we are here 3 years later trying to resolve that problem. And it's interesting that mRNA vaccine that can actually be produced in a container truck. So that's also an interesting lesson learned from me. And there's an increasing global South-led partnerships, such as the Bright Fund, that I think we should look forward to, that the global South countries can actually prioritize our own diseases and our own product development in the next future. And And the other side of the coin is we need to ensure the biosecurity and biosafety in the increasing technological advancement in product development. And BPOM, I think, is responding quite quickly by having the schema OPB, the OPB scheme, and then there's an acceleration scheme that matched the 100 days with CEPI for the industries that invest in Indonesia and conduct clinical trials in Indonesia. And from Dr. Mubagisi, we also see that the pandemic agreement that's been agreed in May 2025 also ushered the phase 2 of the mRNA tech transfer program from the WHO. And we shall see whether we will move forward from the potential to real impact as Dr. Mubagisi has expounded. And from the CEPI, we also learned that even though there are challenges, there are multiple delivery and products in pipeline. And then there's also new investment in manufacturing technologies of mRNA. So it keeps the hope high that mRNA will be viable and scalable for global South countries, including in Indonesia. And from the Q&A session, I think what's my key takeaways is the regional initiative will play an increasing important role in ensuring the balance between peacetime and then also in the pandemic context. With that being said, I thank the panelists for sharing their perspective and insights, including Dr. Martina joining us online. Thank you so much, and I wish you the best for the rest of your day. I know it's still early in your place. Thank you, and see you in the next occasion.
Okay, ladies and gentlemen, please give a round of applause to our moderator and speaker today. Okay, and to moderator, thank you so much to moderator and all speaker. We kindly request all speaker to remain on stage as we proceed to give appreciation after this. Saya mohon Bapak Ibu tetap berada di atas panggung, kita akan ada pemberian apresiasi. So I would like to invite Mrs. Indri Ruslamiyati. Saya mohon dengan hormat kepada Ibu Indri Ruslamiyati selaku Kepala Balai Besar Biomedis dan Genomika Kesehatan untuk dapat memberikan apresiasi kepada moderator dan juga speaker kita di sesi pertama ini. Okay, thank you so much. Okay, 3, 2, 1. Alright. Baik. 3, 2, 1. Oke, dan yang terakhir. 3, 2, 1. Mungkin untuk foto bersama ya. Ibu Indri dengan kedua speaker dan juga moderator kita. Oke, berikan senyuman yang terbaik. 3, 2, 1. Berikan tepuk tangan sekali lagi Bapak Ibu untuk speaker dan juga moderator kita di sesi pertama ini. Terima kasih banyak. Kami persilahkan untuk dapat kembali lagi ke tempat. Thank you so much. We kindly return to your seat. Oke, baiklah Bapak Ibu, tadi kita sudah menyaksikan paparan materi di sesi pertama diskusi kita hari ini. Selanjutnya kita juga akan langsung memasuki pada sesi panel diskusi yang kedua. Namun, sambil kita memasuki sesi berikutnya, nanti petugas kami akan menyajikan makanan dan minuman yang dapat Bapak Ibu nikmati sambil kita memasuki ke sesi diskusi panel yang berikutnya. Dan tema kita di sesi berikut ini adalah Translating mRNA Science into Scalable Manufacturing, Technologies and Future Innovation. Tentunya sebagai moderator, kita juga sudah kehadiran moderator kita di sesi kedua ini, yaitu Ibu Indri Ruslamiyati, MSc APT, selaku Kepala Balai Besar Biomedis dan Genomika Kesehatan. Berikan tepuk tangan yang sangat meriah Bapak Ibu semuanya untuk moderator dan juga speaker kita di sesi kedua ini. Terima kasih.
Oke, baik, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang menjelang sore Ini waktu yang cukup berat ya, semoga nggak ada yang tidur. So I say this is a tough time, so hopefully no one will fall asleep. So I would like to invite all the speakers. I will have 6 speakers that will give us the insight in regards with the translating and mRNA science into scalable manufacturing, technologies, and future innovations. So I will invite the first speakers from University of Indonesia, Professor Dr. Anda Betty Ernawati Dewi, PhD. Please come up to the stage. And the second speaker will be Christian Karol Saputra M. Biotech, is the head of Innovation Center PT Etana Biotechnologies. And the third one will be Ibu Dr. Anda Yuliana Indriati, APTMM. He is the Director of the Research and Development of the PT Bio Farma. And we have 2 our speakers online. The first one is Mr. Rajinder Kumar Suri, is the CEO of the Developing Countries Vaccine Manufacturers Network, or DCVMN. And the second speaker, who is also available online, is Mr. Jose Castillo. So he is the CEO of the Quantum Biosciences. And the last one, I will invite Dr. Chunlin Sin, the Vice President of External Innovation Casino Biologic Inc. So please everybody to, where is Mr. Chin? Okay. So I think he will be on the stage right away. So I think we will invite the first speaker. So Professor Dr. Betty Ernawati Dewi is an expert in infectious diseases. I think she has already lots of research in regard, and also the collaboration research with LPDP, NWO, KISTI, and the Ministry of Health, including the development study for the vaccine mRNA for the dengue tetravalent. So without further ado, so I will give the floor to Professor Betty.
Thank you very much. It is an honor to be here. I will speak about the academic innovation and translational research in mRNA technology. Actually, what I want to talk here is about our own experience about the development of the dengue vaccine. So maybe we will go to the dengue a lot, talk about the dengue. So I'm Betty Ernawati Dewi from University of Indonesia. Actually, this what I will present here is a collaboration research with the National Research and Innovation BRIN, and also from the other university in Indonesia, and we also collaborate with the Etana Biotechnology Indonesia with the support from the LPDP grant. So as previous speaker talk about the mRNA, the academic innovation driving the field about the mRNA actually more on the idea, the new idea. So it should be novel antigen design that can be, the strategy at the conceptual level in the first time, of course. And you know that when we construct that, we cannot go just on the virologist field, but it also goes to the someone who really expert in the biotechnology, in the material science, in the structural biology. So it is need a lot of expert, and also in our study also including also the clinical expert on the dengue research from the internal medicine, and also from the some on the from pediatry. So of course in the academic innovation it's like we want to move the high level of the improvement in the mRNA structure, in the delivery system, and also the formulation, and it's quite tough also to develop that, and we hope that the university spin-off will be spin-off, and then also for the incubation supporting mRNA innovation. And we know that the translational pathway actually started from the discovery and then moved to the preclinical and clinical, and also the application. So in the part of the academic, we will go for the new finding in the academic level and then move toward preclinical and clinical evaluation. For example, for the research in the development of the vaccine, therapeutic, and also diagnostic. And we know that the regulation consideration is really important in the translational research. And of course, you know that I already mentioned before that I only work on the vaccine. So what I want to talk about in here was like development of the dengue vaccine. As we know that the dengue is still a serious problem in Indonesia, and we know that started in the first time that it's reported in 1986, and I mean in 1968, it's the increase of the cases in Indonesia is quite sharply, and also we can see that there is a like cycling when we have the peaking every 6 until 9 years, and the number of cases in this until October in this year is goes to the more than 130,000, and we know that 500 and then more than 500 people pass away by that. And we can see in here, in comparison with other country in the world, oh, I'm so sorry. So maybe this one. Oh, maybe I cannot point it. So actually the case fatality rate in Indonesia is the highest in the world, even though maybe I'm thinking that it's the May of the underreported cases of dengue fever in Indonesia, but we mentioned again that until October 2025, we still have more than 500 people pass away, so it's still a big problem in Indonesia. So already, you know that already many various of the effort that already conducted, including Wolbachia, and also in some that we already have a vaccine also for the dengue, and we recently also developed the NS1 diagnostic, because we're thinking that early detection will save the life of the people. So we conducted the research about how to treat the people more than 2 days of fever, and they never goes to the severe cases. And the NS1 test, yes, rapid test also will prevent of the transmission of the dengue virus itself. But in Indonesia, when I study in many places, that we know that almost of people in Indonesia is like 61%, more than 61% goes to the health facility more than 3 days of fever. So it means they already goes to the critical phase. So it means that quite difficult to save them. That's why we thinking that vaccination is offer the way to ensure that they carry their own protection. So we have the collaboration, as I mentioned before, with BRIN and many researchers of the dengue vaccine in Indonesia, and then we also collaboration with the Tsinghua University under the Etana Biotechnology Indonesia. We try to develop the vaccine. Of course, the vaccine that we developed in here was really want to improve from the current vaccine. So this is the innovation. We're thinking that we really consider about the halal in the mRNA production. And then also we try to use the mRNA vaccine as the modern vaccine platform. Learn from the COVID-19 is better in the efficacy, and using the Indonesian virus strain. But actually, we also developed the artificial intelligence in vaccine design, because we're thinking that even though the based on the strain from Indonesia, we also want to cover almost all of the dengue virus in the world. So we're using the artificial intelligence in the design vaccine. And you know that, of course, there is challenges in the dengue vaccine development. As we know, we have 4 different serotypes with the more than, with the 21 genotypes. So it's become like more various of the dengue viruses. And then there is an issue about the antibody-dependent enhancement that also faced with the development of the vaccine. And the difference in immune responses between the person, between the age, between the gender, and etc. It's become like the key, challenges in the vaccine development. And also the lack of the reliable animal model. So there's some, they already saw the symptom, but it's only showing the severe of dengue, but they never goes to the mild of dengue. So because in the human, they started with the asymptomatic and then goes to the mild, and then there is a severe of dengue infection. And before I talk about the new platform that we know that it's more effective in the dengue vaccine development, now I will give you a little bit explanation about the track record that we already developed in the dengue vaccine. Started with the inactivated dengue vaccine. We do several of the inactivated substance to know the antigenicity and the immunogenicity of the vaccine candidate. And then we also study about, we develop also the DNA vaccine. In here we're using the 4 serotype also, and we can see in here they're able to induce the the antibody neutralizing, and then we also that when we conduct the challenges in the mice that we infect with the cell that infected with dengue virus, the viremia, and then in immunized mice we can see the low level of the viremia. Based on the DNA vaccine, we actually quite easy to move to the RNA vaccine. And we also, in the Consortium Vaccine Indonesia, we developed the recombinant protein vaccine, pre-MA, but University of Indonesia only goes to the dengue serotype 2, and we can see in the sequence that we use it to develop of the recombinant protein, we see that they show the viral-like particle. And now we think that the best vaccine is actually the modern vaccine, because it will be speed. We have the speed that previous speaker already talked about that. Of course, rapid design and development, and also we can see that they also show the strong immune responses, not only B cell, but B cell and also T cell responses. And with this flexible platform, we just change its gain of interest, and with the other microorganism, we will get the new vaccine. And then also that scalability, it is easy to make as long we already established the construct, it's quite easy to make a large product. And also safety profile, as previous study that we using the DNA vaccine, that the demo goes to our nucleus and is integrated with our DNA, but for the mRNA, they're only in the cytoplasm, that it is quite safe for our health, and lower cost over time, of course. And then the recent milestone in the vaccine development that I thought to you that me is only a PI, so actually we have a lot of group from BRAIN and from other researchers. So here I will talk about the milestone of the vaccine development. Of course, we always, every year we do molecular epidemiology to know the new strain that's circulated in Indonesia. And then we using the bioinformatics system, we also try to see the mutation analysis between the candidate of the mRNA vaccine with the new strain that circulated in this year. And we also, the most important also is like we isolate the new viruses, because you know that after we have the construct, we have the mRNA, and then we have immunized mice, we also have to challenge with the severe, with the new virus strain that circulated in Indonesia. So every year we try to isolate the virus for all serotype. And of course, now that I mentioned that it is the innovation from the development of the vaccine, actually we're using the artificial intelligence. Of course, in here is I have to work together with the big data center from Faculty of Medicine, University of Indonesia to develop this, because it is out of my knowledge. So, but I have the idea about how to design the vaccine using this to predict that the vaccine will still work even though the dengue virus already have mutation as RNA viruses. So we're using artificial intelligence to design this. So it's quite tough because we have more than 100,000 data to analyze that. And then we also using the artificial intelligence to predict of the dengue transmission and then of the dengue cases. In some places, it is also a good strategy to have a strategy for the vaccination in the future, because in here we also evaluated the antibody that have in the certain area in our study, because we also have in many places in Indonesia. And this is the one that we conducted with construct of the dengue vaccine, and of course should be tetravalent, but we have modification because you know that the main problem is like antibody dependent enhancement, so we try to modify it of the gene to avoid the antibody dependent enhancement. And this is, we have collaboration with Tsinghua University, and of course we hope that we can launch the product in this in the following years, in early years. And this is the antibody is already, antigen already induced the antibody, and then we still analysis for the next about the ADA of the candidate. And this is so the modification of the antigen that the mRNA vaccine that is already produce the protein, whether they really change of the mutation that we want to avoid of the ADA. It is in dengue 1, 2, 3, 4. I cannot tell in detail. And of course, you know that the research about the vaccine is not only we produce the vaccine, but capacity building is more important because I may be getting old, so I really need somebody who doing research in the new future. So in this study, I involved like undergraduate student, graduate student, from the master student, and also from the doctoral degree to continuing of our study, and they have also experienced the good research in Indonesia. So as a conclusion, actually the development of mRNA vaccine technology reflect of the combined effort from the academic research, interdiscipline, and also institutional support. And modern platform vaccine, we include the mRNA technology that also involve the artificial intelligence can produce the good vaccine in the future. And the collaboration with other institutions and the university that have experience in the production of the mRNA vaccine, it's really a good, to improve of our production from the academic side. So I think that's all of my presentation. So I really would like to thanks from the LPDP that already support us in the research, and also from my friend, all of dengue research vaccine team, and also from Etana Biotechnology, Brin from the education, and from China, from Tsinghua University. So I think that's all because of the time. Thank you very much for your attention. Thank you.
Okay, so thank you very much, Professor Betty. I think it's 15 minutes sharp. So thank you for finishing on time. So I think this session is kind of different from the previous session. The previous session is more about the scientific foundations as well as the global frameworks. But this session is more to the practical realities of developing, producing, as well as innovating the mRNA technologies at scale. I think it's already proven by Professor Betty's presentations. So I know all of you now, everybody is listing questions to Professor Betty, so please keep those questions. So we will move to the second speaker. So Pak Christian Karol Saputra, Head of Innovation Center from PT Etana. He will speak about the local manufacturers of the mRNA vaccines, lesson learned from the COVID-19. So please, Pak Christian, the floor is yours. You have 15 minutes. Okay.
Okay, so thank you. So first of all, I would like to say thank you, and then I would like to say permission because I would say the presentation in Bahasa. Most of it is from Indonesia, so I would like started the presentation in Bahasa Indonesia. Okay, so okay. Jadi saya ingat pada waktu saya join ke Etana 10 tahun yang lalu, saat itu belum ada industri biotek. Hanya ada satu, which is itu Bio Farma. Dan saat itu saya yang kebetulan juga dari salah satu manufacturer chemicals, move to biopharmaceutical, which is itu Etana, belum tahu apa itu produk biotek, dan kita harus belajar mengenai produk biotek itu sendiri. Dan kejadian ini terulang lagi ketika pandemi COVID-19 itu datang. Which is saat itu vaksin mRNA, salah satu terobosan tiba-tiba muncul dan orang belum tahu apa itu vaksin mRNA. Sehingga kita harus dengan cepat beradaptasi mengenai vaksin mRNA itu. Jadi banyak hal yang pada akhirnya menjadi tantangan, challenge bahwa apakah itu safe, apakah itu akan berefek, apakah itu nanti bagaimana dengan long term study-nya, bagaimana jika dia terintegrasi dengan DNA kita, dan sebagai seorang apoteker yang terbiasa dengan obat-obat chemicals dan saat ini harus menghadapi obat biotek, itu menjadi suatu tantangan yang sangat besar. Sehingga pertanyaannya, apa itu mRNA? Kenapa kita harus menggunakan mRNA instead of kita menggunakan yang biasanya? Bukan kayak biasanya yang inactivated sudah lebih terbukti gitu kan. Dan banyak sekali mulai dari where, when, dan segala macam pertanyaan di benak kita yang pada waktu itu belum banyak orang bisa menjawab segala macam pertanyaan terkait dengan mRNA vaksin. Jadi Bagian mana yang paling sulit? Bagian tersulitnya adalah taking the first journey. Jadi bagaimana kita melangkah untuk pertama kali untuk say, yes, Etana mau coba kita jalan dengan vaksin mRNA. Saya, apoteker, pada waktu itu masih sekolah studi S2 Biotek, which is belum lulus, gitu ya, masih ongoing studi. Dan saya harus mempelajari at the first, apa itu mRNA, dan bagaimana kita belajar bareng bersama regulatori, bersama dengan seluruh tim Etana, taking the first step untuk mencoba sesuatu yang baru. Jadi itu adalah the hardest challenge. Mengumpulkan semua keberanian dan meyakinkan semua orang bahwa kita harus melangkah maju. Oke, jadi apa saja yang sudah kita hadapi, tentunya bagaimana strategi Etana dari transfer teknologi untuk masuk ke Indonesia melalui transfer teknologi tadi, sehingga Indonesia nantinya akan memiliki building capacity yang cukup baik. Oke, ini adalah track record dari start awal sampai akhirnya kita selesai melakukan proses produksi vaksin mRNA. Dan major challenge apa saja yang kita lalui pada waktu itu? Oke, saya ingat betul, jadi pada waktu itu regulasi bahkan belum ada. Regulasi dari global pada waktu itu bahkan dia masih dalam model revision. Belum yang regulasi yang sudah fix. This adalah regulasi yang terakhir dan terupdate. Semua masih berupa regulasi yang draft version. Sehingga tidak ada sesuatu regulasi yang kita bisa pegang. Kemudian dari human dan organization capacity building. Waktu itu mRNA masih tergolong sesuatu yang baru. Sehingga orang yang mempelajarinya pun bagaimana kita meng-hire orang, itu cari orang susahnya setengah mati. Bagaimana saya menginterview berpuluh-puluh orang dan yang ngerti terkait dengan mRNA itu hanya beberapa. Kenapa? Karena ini adalah situasi yang unik, kombinasi antara farmasi dan juga bioteknologi. It is not pure farmasi, tetapi juga not pure biotech. Jadi harus kombinasi keduanya. Oleh karenanya bagaimana kita harus membuild kapasiti dari orang itu menjadi suatu challenge lain tersendiri. Kemudian juga dari segala macamnya bagaimana shortage semua reagent, semua supplier shortage. Bagaimana kita tidak bisa melakukan pembelian secara langsung. Bagaimana mereka tidak bisa melakukan pengiriman ke Indonesia karena produksi di negara asalnya pun mereka tutup dan shortage. Sehingga shortage pun segala macam reagent, segala macam excipient itu kita juga tidak miliki at that time. Jadi harus dengan segala macam resource yang ada kita berpikir out of the box untuk bagaimana proyek ini dapat berjalan dengan sesuai dengan timeline yang pada waktu itu kita harus rush begitu ya. Oke. Dan termasuk bagaimana regulatory challenge tadi, bahwa regulasinya masih baru, sesuatunya masih baru, berubah-ubah terus dari global dan yang versi Indonesianya pun bahkan belum ada, sehingga kita mau nggak mau harus mengadap versi global yang kita harus pantau secara berkala. Kemudian supply chain resilience, jadi oke, secara fasilitas kita juga harus catch up, tetapi di sisi lain bagaimana dengan reagent yang harus kita beli sesegera mungkin. Kemudian bagaimana dengan semua media, bagaimana dengan semua consumables yang harus juga kita lakukan pembelian dan mendatangkan sesuai tepat pada waktunya. Itu adalah suatu huge challenge. Tidak hanya untuk bagian ekspor impor, tetapi juga dari bagian user dan juga ke government. Kenapa? Karena kita harus meminta push balik ke government. Bagaimana untuk mendatangkan proses ini dengan cepat. Kenapa? Karena saat itu restriction ada di mana-mana. Kemudian internal capacity building dari kita. Kemudian robustness proses. Once semua kebutuhan sudah ada, maka kita harus mendefine suatu proses yang robust, yang ketika diulang akan menghasilkan suatu produk dengan tingkat keberhasilan yang tinggi, dengan yield yang baik, dengan quality yang baik. Itu juga merupakan suatu challenge tersendiri. Jadi ternyata it's not that simple at that time. Dan tentu kita tidak ingin mengulangi hal itu terjadi lagi. Dan once suatu pandemik itu datang, kita berharap kita sudah siap. Tapi pertanyaannya, apakah kita hanya mempersiapkan diri untuk pandemik atau sebenarnya dari platform mRNA ini kita bisa menyiapkan untuk banyak hal yang lain. Maka kita bisa lihat di sini, ternyata mRNA itu sebenarnya adalah sebuah platform, sebuah delivery system yang tergantung kita mau menggunakan untuk apa. Kita bisa menggunakan untuk sel terapi, kita bisa menggunakan untuk protein replacement, kita bisa menggunakan untuk vaksin seperti yang sudah kami kolaborasikan dengan UI, dengan Prof. Betty, kita bisa menggunakan untuk genome editing. Jadi mRNA itu adalah sebuah platform, sebuah kendaraan, tergantung kita mau menggunakan itu untuk apa. Saat ini mungkin kita semua berpikir berbondong-bondong ngerjain semuanya vaksin. Padahal di sisi lain BPJS kita, Kementerian Kesehatan kita, mereka punya heavy burden di penyakit-penyakit yang lain yang sebenarnya bisa kita bantu dengan platform yang sama yang saat ini sudah dimiliki oleh Bio Farma untuk kuantum tadi. Jadi pertanyaannya, apakah kita akan fokus satu semua di vaksin atau sebenarnya kita akan mengutilisasi platform mRNA itu untuk berbagai macam pengobatan yang lain. Dan kita bisa lihat di sini, oh ternyata banyak loh kita bisa gunakan untuk berbagai macam disease yang lain yang mungkin kita belum berpikir out of the box saja. Sama seperti ketika COVID, pada waktu itu kita dipaksa untuk berpikir out of the box. Saat ini fasilitasnya sudah ada dari government, orang selalu komplain mana interaksi antara akademia, bisnis, dan government. Yet, saat ini kita di sini hadir, kenapa kita tidak berpikir out of the box? Oke, Univ A, Univ B, Univ C sudah ngambil vaksin, dan saya ngambil di area yang lain. Let's say saya ngambil genome editing, let's say Univ C ngambil protein replacement, atau bahkan mengambil bagian sel terapi. Jadi bisa banyak dengan platform yang sama, isi berbagai macam, bisa menghasilkan obat yang banyak yang bisa mengurangi burden dari BPJS yang saat ini sedang kita hadapi bersama. Jadi bisa teman-teman bisa lihat di sini, dari R&D, manufacture, dan regulatory, semua memiliki challenge masing-masing, dan dengan sistem mRNA plug and play, ini akan mempermudah banyak sekali hal. Mulai dari design cycle, proses development, GMP validation, termasuk paling penting adalah adaptability. Jadi bagaimana design cycle, oke, kalau saya mau melakukan pembuatan let's say sel terapi ataupun terapi yang lain, kita cuma tinggal ganti isinya, tinggal ganti ininya dengan model desain yang sama, formulasi yang sama, then it tadi dalam waktu 100 hari aja udah bisa melakukan uji preklinis. Maka pertanyaannya harusnya itu juga bisa dilakukan untuk penyakit-penyakit yang lain. Maka kita bisa bergerak bersama untuk mengurangi burden yang saat ini ada di kesehatan kita. Jadi ini di sini bisa terlihat bahwa potensi dari mRNA based terapi ini sebegitu luasnya, tidak hanya vaksin. Mungkin saat ini kita berfokus di preparedness, saat ini kita berfokus di bagaimana untuk vaksin, tapi sebenarnya ini adalah sebuah platform yang memiliki window lebih dari itu. It's not just about vaksin, tetapi dia punya berbagai macam potensi yang saat ini mungkin kita belum pikirkan, belum gunakan, atau bahkan kita belum terpikir, oh ternyata si mRNA sistem delivery ini bisa digunakan untuk penyakit ini juga ya, oh ternyata di luar negeri bisa dipakai buat ini juga ya. Jadi ngapain kita sama-sama mengerjakan sesuatu, oh semua ngerjain vaksin, kenapa kita enggak mencoba berbagai macam angle sisi yang lain. Kalau sudah ada yang ngerjain vaksin ini, ya berarti saya ngerjain sel terapi yang lain atau protein terapi yang lain. Jadi bisa berbagai macam angle yang kita bisa gunakan bersama-sama. Jadi ini adalah current update terkait dengan mRNA based drug therapy. Jadi bagaimana ternyata sudah banyak sekali bahkan yang sudah sampai di uji klinis. Yang tentunya ini terpublish Dan kita bisa melihat bagaimana sekuensinya, bagaimana dia desain model mRNA terapinya, bagaimana dia desain LNP-nya, yang nantinya kita menggunakan reverse vaksinologi dengan reverse teknologi kita gunakan ulang dan kita gunakan untuk di Indonesia untuk penyakit-penyakit yang saat ini mungkin memiliki burden yang sangat besar terhadap BPJS kita. Termasuk kalau dilihat di sini global untuk infeksius mostly terlihat bahwa banyak sekali untuk berbagai macam termasuk vaksin flu dan segala macam, tapi sebenarnya lebih dari itu tidak hanya hanya berfokus di vaksin, tetapi bisa lebih dari hal itu. Dan
2 menit
bahkan
lagi,
beberapa
Pak.
sudah vaksin.
Ya, 2 menit
Oke.
lagi.
Nah, jadi pertanyaannya apa saja yang sudah dilakukan? Sudah banyak kolaborasi, salah satunya dengan tadi Bio Farma, kemudian juga dengan Etana, bagaimana Indonesia dan WHO dengan expert vaksin production. Jadi banyak sekali kolaborasi dan sekarang pertanyaannya adalah kita semua di sini tinggal ayo kita mulai kolaborasi tersebut. Tidak hanya sekedar berbicara, tidak hanya sekedar ide, tetapi kita mulai kolaborasi tersebut. Tentunya Bio Farma di sini tadi saya juga udah barusan ngobrol dengan teman saya yang baru minggu lalu ketemu, begitu ya Bu Indri. Jadi minggu lalu kita baru ketemu, kita ngobrol, jadi pertanyaannya saat ini sudah ada dan sudah disediakan pemerintah, maka tinggal kita memulai suatu langkah kecil itu sama seperti ketika Etana memulai langkah kecil pertama beberapa tahun yang lalu. Kurang lebih itu, terima kasih.
Oke, terima kasih Pak Christian ya. Jadi tadi paparannya jelas menjelaskan tentang how important the first step is taken, because I think after we move to the first step and the other step will follow, and also the challenges that already faces by the Etana at the first for the COVID-19 pandemics. So we will move forward to the third speaker. Pembicara ketiga kita adalah Ibu Yuliana Indriati, Director of Research and Development for Vete Bio Farma. So she will speak about the developing of RNA vaccines for emerging pathogens, lessons from the MERS development. So silakan Bu Indri.
Yes, thank you.
Namanya sama nih
Thank you, Bu.
kebetulan.
Okay, thank you for time for the Bio Farma to share about the our experience in starting the mRNA development. At the first, maybe this is the background of the Bio Farma. Actually, Bio Farma already focusing in the vaccine starting more than 130 years ago. And then actually our capacity is very huge because our capacity every year we produce 3.1 billion doses. And the vaccine that we are producing from Indonesia, actually we already export to globally because we have some of the vaccine that already get the PQ WHO. This is actually about the positioning of the Bio Farma in the global. Actually in the volume we are number 7 in the rank of the global that we are distributing globally, and Indonesia itself we are number 1 with the market share is more than 95%. About the Bio Farma vaccine journey, actually Bio Farma supporting for the national immunization program, and also Bio Farma active in the making awareness in the making of the vaccine. So that's why actually Bio Farma also contributing in the expanded program on immunization. So it's meaning that Bio Farma also participate in reduce mortality of the children almost more than 40%. Every year that we are producing the vaccine, actually after we are market the vaccine, then we are actively support the Ministry of Health in making the awareness of the using in the vaccine on the target customer, or also in the clinical practice of using the vaccine. This is the vaccine that we are producing locally. Actually, almost all of the vaccine that mandatory from the Ministry of Health, we are producing locally. However, for the vaccine, some of the vaccine we are in-house R&D development, and some of the vaccine we are doing the tech transfer. So it's meaning that the vaccine that needed in the Indonesia, actually Bio Farma would like to contribute with the importation first while we are doing the in-house product development. And then the technology vaccine on the technology platform, actually Bio Farma already covered the expertise of the live attenuated, inactivated, virus-like particle, protein recombinant, and also in the conjugation. And the last of the technology platform that now we are starting to build our expertise is in the mRNA and viral vector. This is the background why Bio Farma in developing the mRNA, why we put the antigen like MERS or other antigen in the pandemic preparedness, because Bio Farma thinking that the preparation of the pandemic preparedness is very important, because if we see about here, actually when we have a pandemic of the COVID-19, we are making the development almost 1 year. If we can make the readiness of the vaccine in the 100 days of the mission days from the CEPI, actually the saving of the life of the population we can decrease into 8 million. So this is the way that we are following with the CEPI, with the Indonesian Ministry of Health, why we are making some of the antigen in the mRNA or viral vector. Because of the platform itself, we can accelerate the development to prepare of the pandemic preparedness for the mRNA, because we don't need to the cell cultivation, and also we can easily in the closed system with the quantum, we can upscaling more faster compared with other platforms such as like the protein recombinant or live attenuated or inactivated also. And in preparing the pandemic preparedness, actually we make the integration between the technology that we choose and also the readiness of our facility, and then the supply chain and regulation itself to make the readiness of the antigen of the mRNA in the phase 1. This is actually the slide the same with Ibu Rizka previously. Actually, this is the journey of the Bio Farma in making the mRNA our expertise and also our facility. Actually, it is starting. In the 2021 when the finance minister give the funding to the Bio Farma for making the facility for the mRNA and viral vector. So it's meaning starting 2021, we build the facility for the mRNA and viral vector that actually will be ready to get the GMP certification in the 2026. It's meaning that we are ready for the clinical trial phase 3 using the mRNA in the 2026 and the commercialization actually in the 2027. The journey of the Bio Farma actually at that time we starting to capacity building with the Manchester. We make the collaboration with Manchester University, starting to have the expertise of the mRNA platform. And then continuing with the 2022, we get the tech transfer from the African with the support from the WHO and MPT to get the tech transfer from the African with the COVID-19 mRNA. Then continuing again in the 2023, we get the collaboration with the CEPI and also the funding from the CEPI to make our bigger facility and also the expertise of the Bio Farma. Then Bio Farma buying the MIDI quantum. Using the quantum tensify technology, actually we use the mRNA rabies. And in the 2025, actually we make the collaboration again with the mRNA consortium for the mRNA malaria with Senegal and also with the BioVac. And with the Gates Foundation, actually Bio Farma also get the funding again and also the support from the Gates Foundation to increase the capability making antigen of for the mRNA vaccine for the AI. And also we make the expertise in the pDNA for the raw material of the mRNA manufacturing process. And now the facility that we are building from the 2021, we will get the GMP certification for the our facility for the pilot for the pilot scale for the clinical trial phase 1 and 2. Hopefully it will be get the GMP in the 2026, and also for the bigger building for the clinical trial phase 3 and commercialization. This is actually we get the appointed from the CEPI for the manufacturing facility network. The scheme that CEPI would like Bio Farma to make expertise in the mRNA, then we make the clinical trial phase 1 and also the bigger facility for the pandemic preparedness. We already ready with the facility to produce the antigen mRNA. And the network actually for collaboration for the development, we make the consortium with some of the company and also from some of the countries. The benefit is to accelerate the product development up to the phase 1. This is the background why we enter the MERS, mRNA MERS, because as advised by CEPI, and also MERS is one of the antigen in the WHO priority list. And also if we see here, actually a lot of Indonesian people go to the Saudi Arabia, whereas this case is very high in Saudi Arabia. And then the case make the mortality up to 40% from the cases. And up to now, there is no approved vaccine for this disease. So this is the background why Bio Farma put the MERS mRNA that we would like to develop to be a pandemic preparedness up to the phase 1. If we see about the globally, actually MERS vaccine mRNA, only 3 companies develop. However, Bio Farma only the one that developed with the mRNA platform. The others is with a viral vector or DNA platform. Currently, the progress of the mRNA development for the antigen MERS, we make the co-development with UNPAD, Pajajaran University, and with the support from Gates, because Bio Farma would like to have the expertise in the AI, so we make the more fully optimize the sequence of the antigen. So there are 2 antigen of the MERS mRNA that we would like develop up to the immunogenicity testing. This is the potential that we would like to have a partnership to more development to get the co-development of the mRNA MERS. Actually, we would like to get the partner in making the reference material, and also in the preclinical and clinical, including the regulatory. We would like to have the strategic partnership in this area. And the last, this is about the product pipeline that Bio Farma in our research and development still in the development is in the COVID-19, in the MERS, and also in the rabies, influenza seasonal, and also we would like to enter into therapeutic application, like to the oncology area. Thank you.
Okay, thank you very much from Ibu Indri from the Bio Farma. So I think there's lots of things that have been shared in regards with the efforts of the Bio Farma to produce the vaccine. As we know, the Bio Farma is one of the largest vaccine company, and also their roles and the lessons learned from the MERS-CoV due to globalizations. There are Indonesians that went to the overseas that can cause the MERS-CoV, and also it's also enhancing the collaborations with universities as well as with the funders and also with the academicians. So we can move to our next speaker. So are you there, Mr. Rajnav? Hello? Can you hear me? Mr.
Are you
Rajinder?
addressing to me?
Mr. Rajinder?
Yes, I'm here.
Yes, okay. So Mr. Rajinder is the CEO of DCVMN. He also serving as a committee delegate on the Gavi PPC and also the Gavi's vaccine investment strategies, and also a member of JP. So I think with his experience, he will give a talk about the strengthening the mRNA capacity and capability in the low middle income countries. So Dr. Rajinder, the floor is yours.
Thank you very much, Chair. At the very outset, I would like to thank Director General Dr. Rizka for inviting me to this very important platform and sharing views. So thank you very much for that. And also, I would like to thank Honorable Minister Budi, who set the tone this morning for, by laying the vision for Indonesia, as also what it means for each country to really think about and what is required to be done. So, I have very fresh memories of having conducted annual general meeting of DCVMN in Bali, Indonesia, last month, and this is something which is coming as a great booster, having really experienced the hospitality and the kind gestures of team from PT Biopharma as also from Government of Indonesia. So with these words of thanks, I would like to open my presentation, and I hope you can turn the slides. So the next slide, please. Thank you. So I'll go a little broader. Let us look at the geopolitical challenges and what are the global health challenges that is facing the whole world, and then we'll narrow down to mRNA. So as far as geopolitical challenges are concerned, you have seen the pandemic agreement has now been signed, so it's a very big achievement with 194 countries coming to consensus and signing the agreement, thanks to WHO and the whole secretariat who was involved in INB discussions. And now, of course, the annexes are being discussed, debated, and thrashed out so that all the annexes are also available, particularly pertaining to Article 12, which deals with the pandemic preparedness. Now, we have witnessed that the US administration decision to withdraw funding from WHO definitely has had a spiraling effect, and it is not only the financial impact that we are talking about, but it is also on the technical and scientific inputs which were vital for rapid response to any public health emergency of international concern. And I think this is a matter of real concern to all the countries in the world. Talking about the global health program disruption, you have seen that there is the loss of funding for critical programs like HIV, AIDS, tuberculosis, malaria and polio will get impacted because US was contributing almost 12 to 15% of the WHO's budget. And more than that, it is also the global monitoring capacity, the ability to detect and respond to any emerging health threats may be compromised. On the other side, in June, during the replenishment meeting of Gavi, what happened was that there was a communication from the US administration withdrawing support even to Gavi for funding, and there is a huge gap of $3 billion which may have a serious impact on the vaccine procurement budget, because it was $7.6 billion budgeted in the original plan, but now it has gone down to 5.9 billion. So all these things are going to definitely impact. Next slide, please. So, this is also leading to some global disparities because there is lack of timely access to novel technology. mRNA, we have been talking right from, this afternoon. You have been witnessing the learned speakers are talking about mRNA, but during COVID-19, there was a very, very big limitation to access to the technology of mRNA, and it was not available for LMICs. And then there is a lack of infrastructure when you're talking about, for example, laboratory network for characterization or neurovirulence test, in non-human primates. It is a big challenge in our countries. Third, of course, is the regulatory uncertainty, because the shifts in healthcare policy can deter investments, and then leading to inflation and interest rates volatility, which can have huge impact because of the economic instability. And of course, all of you are familiar with the fragmented global supply chains, which, despite efforts to localize many critical inputs, still depend on fragile international networks. There was a high dependence on foreign supplies, particularly single-use disposables were only being manufactured in few countries, very few countries like Germany and US, and during pandemic there was no availability of these inputs, bringing to a grinding halt. Next, please. Next slide. So when we talk of global challenges, you have seen in terms of funding squeeze and the market uncertainty, because there is a donor fatigue and tighter public budgets, which are becoming more and more common now, and it is not only US but many other countries are also pulling back from the budgets. Procurement validity reduces the market visibility for manufacturers, and of course the intellectual property and access is becoming a big issue. You heard many honorable speakers today about technology transfer, which is helping in terms of the manufacturing capacity, and the regulatory harmonization, which is going to be a critical factor. Next, please. So I think ultimately it is the funding imbalance which is a priority, because today, the challenge faced by low middle income country is balancing. It is balancing between quality assured medical products meeting expectations and versus accessibility, affordability and availability. So these factors are ultimately deciding the public health good, what ultimately we are here for. Next slide, please. Next slide, please. Yeah. So what are manufacturers looking for to build and sustain this scalable, MCM, the medical countermeasures capability? Of course, we are definitely looking at policy stability and predictability. We are looking at public-private partnerships, the flexible manufacturing platforms, supply chain resilience, and digital transformation. This, is going to be the key. Next slide, please. Access to novel technology, and here I would like to thank WHO, MPP and Afrigen that mRNA technology has now been made available to 11 manufacturers in low middle income countries through this hub and spoke model. IP protection is continuing to be a big issue, because clear legal frameworks and guardrails are essential for making sure that the manufacturers are protected. Regulatory alignment, of course, is because transparent and predictable regulatory environment is essential to reduce risk and delays, which can otherwise create, as Dr. Sushant was talking about, it takes 7 years for a product to see the day of light of the day, and then another 5 years if there is any label change. Infrastructure and workforce, I think, are going to be critical factors, whether it is in one country or the other. Unless you have the trained workforce, unless you have electricity, airports and roads and everything else to make sure that the technology is being reproduced. Next, please. Now, talking about mRNA technology, you heard many learned speakers on this part, so I will just say that mRNA is the latest platform in vaccines for infectious diseases with advanced technology incorporation, precision immune response, enhanced safety and faster response time with an ever-evolving scientific landscape. So this is something which I think is the future. But at the same time, a word of caution that let us not put all eggs in one basket, and let's not think mRNA is the only panacea for all your problems. That may not happen. Next, please. So, what are the challenges in mRNA? And I think some of these challenges were mentioned by some of the panelists, but at the same time, heat is the enemy of mRNA supply chain because ultra low temperature storage is required -70 degrees or even lower for long-term stability, and lack of infrastructure, which I just talked about, because unless you have the deep freezers, equipment everywhere and reliable electricity, it is going to be a big challenge. And then there are breaks in the cold chain, there are high transportation costs, there is a manufacturing complexity. Everything is not simple because these are 200 components in one vaccine that you need to ensure and procure. Next, please. So there several initiatives have been taken by our member companies. This is only a snapshot of some of the examples. Like Genova is, they have brought out an innovation, which is a platform technology advantage. It's a self-amplifying mRNA for a long-lasting immune response. And as you know, this is 2 to 8 degrees stable, unlike many others, which were rather the two vaccines that were brought out during COVID-19. And they have the capacity and a scalable process to take it to 1 billion doses per annum, and the technology is available for tech transfer. Synergium Biotech from Argentina, has been leveraging the WHO and MPP mRNA technology transfer H5N1. And likewise, PT Biopharma, just now you heard my colleague talking about the PT Biopharma program fully supported by CEPI, and then likewise, Manguinho Fiocruz from Brazil has also been benefited with this technology. What is common among all these and many others is that there is WHO MPP support, that there is a CEPI coming fully behind these, and then there is Quantum, who is strolling around among all these manufacturers to make sure that this technology is fully absorbed and taken up very quickly. Next slide, please. So, several international initiatives, and DCVMN has been supporting regional vaccine manufacturing. As you heard, WHO mRNA Technology Transfer Hub, which has already made a great contribution, and CEPI, R&D and capacity building. In terms of Gavi, AVMA, the African Vaccine Manufacturing Accelerator has been introduced. It's a $1 billion fund. And then IFC and Regional Vaccine Manufacturing Collaborative and Asian Development Bank have been coming forward for several other support. And DCVMN has been contributing in terms of workforce development, in terms of industry representation, technical capacity building, knowledge exchange, and healthy industry framework. Next, please. So there are three big roles that DCVMN plays today: is advocacy, both in terms of voice share and wallet share for member companies, partnerships, whether it is access to novel technology, R&D, tech transfer, scale up and scale out, and workforce development. Next slide, please. So you can see here there are 46 member companies in DCVMN with 8 working groups and 1 technology transfer training delivered, 2 VR virtual reality programs delivered, and 2 quality by design and manufacturing science new initiatives have been launched, and 3 publications. Next, please. Next, please. So these are some of the actions that DCVMN is taking in terms of healthy industry framework, that we bring people together and make sure that they get access to technology, finance, and everything else. We have these working groups, including intellectual property, where we can support manufacturers, and we are seriously working on the technology transfer training. Next, please. I'm very happy to share with you that DCVMN members have forged 116 collaborations between 2021 and 25, and out of these South-South collaborations are 41, and with international organization another 41. So you can see the amount of credibility that you gain by being member of DCVMN. And next, please. So this is the strategy for DCVMN: sustainable manufacturing, sustainable financing, and workforce development. And with these few words, I rest my presentation. I think I am well in time. Thank you.
Thank you very much, Dr. Rajinder from the DCVMN. I think the roles of the DCVMN is to strengthen the global vaccine supply and access. I think we are quite interested with the wallet share that you mentioned with the punctual punctual marks there. So for the low middle income countries. So yeah, so please keep your questions for Dr. Rajinder, because like we will move to our fifth speakers is Dr. Jose Castillo. So he is the CEO of Quantum Biosciences. I think we talk about his platform and technology since in the morning. So now we would like to have him to give a talk about enabling innovative technologies for the mRNA research and manufacturing. So please, Dr. Jose, the screen is yours.
Thank you so much. Hopefully you hear me well. So it's a great pleasure and an honor. Thank you
yes, we
so
can
much
hear you
for
really
the
well.
invitation. Super. Thank you so much for the invitation, giving me the opportunity to give you, the perspective of a company that is developing RNA technology. So a company that is not actually, developing vaccines, but trying to provide technologies that hopefully meet, the needs of our partners and customers. Maybe if you go to the next slide, this is a slide that I think is very important. Because what we face, our company was started up in 2021, and since then, so it's more or less 4 years ago, we have seen a very rapid and fundamental change into the RNA market. You see, this is just one of, a long list of available data, one snapshot, where you see the amount of clinical trials using RNA as a modality for infectious diseases and oncology. You see, obviously, 2021, there was more than 100 clinical trials. As you can imagine, most of them were focused on COVID-19, and a big portion of those were sponsored by Moderna, BioNTech, CureVac at the time, and then a couple of others. And since then, of course, RNA has proven to be, probably a good modality for vaccines, especially from pandemic readiness, and also for other vaccines. And there is, several presentations that were given today. But at the same time, there is less need in terms of mass of RNA. And there is, other applications of RNA, and the sector is really broadening, especially in oncology. There was a presentation, given by Biopharma talking about also other type of applications like, gene editing and in vivo CAR T cells, protein replacement and the like. So this is actually a fundamental change in a very short period of time. The other thing that we observed since 2021 is that today when we talk to our partners and customers, what they tell us is that, of course, they are interested in technology to synthesize, purify RNA, technologies for the encapsulation of RNA, also very much interested into RNA design capabilities. But actually, today, what we face is a market, and this is great actually, is a market that is looking for collaborations. Not only to buy machines or buy reagents or buy disposables, but it's more, we are more, in front of in a situation where people are looking for an RNA development partner that hopefully has an end-to-end platform like the one we have developed. But so it's for me it's unique in my experience developing disposable technologies for biomanufacturing. Is such a very fast fundamental change in what the market is expecting, and so this is actually what we are trying to do. If you go to the next slide. So basically, in these 4 years, we developed a platform that is composed of 3 elements. The one in the middle that indeed, as you said, has been mentioned in several presentations, and we are honored actually to have early adopters that trusted us and continue to trust us to, for this tech, which is an integrated, scalable from milligram to kilogram scale. So a technology that incorporates both RNA synthesis and RNA purification. But then, on the left and right side of the slide, we also developed today our own algorithms to optimize the design of RNA, taking into consideration, the antigen or the protein that our partner wants to develop. And so we develop all the constraints that are requested for that RNA to code for the protein at the right time, in the right cell, with the right level of functionality and stability. And on the other side, of course, RNA on that. So if we think that in terms of platform, what we offer today, if I start actually, by the 4th criteria on the slide number 4, so which is the next one, there might be actually a delay between what you see and what I see. So hopefully you see a slide actually with 4 buckets where I develop the benefits. So it's essentially something that we wanted to offer is freedom to operate, which is essential for everyone. So today we have developed our delivery tech, our synthesis and purification technology, guaranteeing, to the extent this can be guaranteed, that there is no patent infringement. Very cost effective. You will see one slide on actually animal health. Cost is important for all, but especially when you develop vaccines for poultry, is actually of the essence. I don't know if you still hear me. Hello?
Yes. Yes. Next slide.
Wait, I hear you. Next slide.
Okay, thank you. Because okay, super. So I was talking about this one indeed. Very cost effective, fast, scalable. So the slide number 4, and indeed de-risked in the sense that what we develop essentially is a process first that delivers on CQAs and on yields, which has been integrated into machines. But so if you go to slide 6, just a couple of seconds on ENCODE. So the
slide 6, please.
Series of next one. Exactly, thank you. So this is something that we have been developing over the 3 last years. This has been used already for more than 200 RNA constructs, 50 of them tested in vitro and sometimes in vivo, depending on our partners and customers. This is, the obvious first step to translate, your antigen or your protein into a functional RNA. So today this platform is actually available, and we offer it only through collaborations because, we are not a software company. So this is offered actually at the very first component of the R&D agreements that we develop with our partners. So if you go to the next 2 on the intensify, so the RNA synthesis and purification tech. So you move ahead to slide number 8. So this is what I was saying, that our process
slide number
is
8.
Today. Slide number 8. Yeah, I'm sorry. Next one. Super. Thank you. So this is actually what I was saying, that we develop essentially a process. And then the process has been integrated into a machine that operates it. So we have now, multiple cases where the process proven to be delivering in terms of yield and in terms of critical quality attributes, both for mRNA and for self-amplified RNA. And we are now in a process to apply for a drug master file in the US, to deliver on consistent quality. On the next one, 9, just a slide to summarize what I was saying initially, from milligram, to kilogram. So manual kits for people, to test that the process delivers, hopefully according to their expectations. R&D processes and equipment in the middle, and the intensified MIDI, which is the system that delivers large quantities under GMP compliant, in a GMP compliant environment. And on the next one, the evidence, slide 10, the evidence that indeed, when you apply actually for a given DNA template, the same process from the manual kit at the 1 milligram scale to the intensified MIDI. In the case here, it's to 1 gram scale, but this one can be scaled up to, to a large scale manufacturing production. What you can see is that from the production perspective and from the CQA, talking about integrity, capping efficiency, and residuals, we see, a perfect similarity between all the scales. And then as time is flying, maybe you go to number 13, so I can talk a little bit about another two. We can talk a little bit about
Slide 13.
Another two. Yeah, another one.
Satu lagi. Nah.
Thank you. Perfect. About actually our encapsulation technologies. And so here again about, the goal is not to go deep into technical details, but maybe to insist again on the IP intricate landscape and our, desire since the beginning of this development to provide freedom to operate. So to achieve this goal, actually we decided to stick to the current paradigm of four component lipid nanoparticles, but where we did substitute the cationic ionizable lipids that provide actually, the great functionality and immune response when you use RNA as a vaccine, by another molecule that we developed for this use that is fundamentally different from the chemical structure perspective, but that was designed to deliver some of the, chemical functionalities of lipids. So this class of molecules called calixarenes is today actually a fundamental component of our four component LNPs that delivers actually functionality, as we will see in two occasions, in two cases, and delivers also freedom to operate. And so this is something on the next slide number 14, coming back to what I was saying at the very beginning of my talk. Of course, RNA is still a great modality, very fast, actually, that delivers, triggers a very robust immune response, certainly designed for pandemic readiness, for the large-scale production of vaccines, but at the same time, there is many other applications, and so here I'm referring to one of them that we published, almost two months ago, with a big hospital center in New York, using this new delivery technology to deliver RNA into their pancreatic cancer patients. So this is one of the applications we are pursuing for this new class of chemicals. And so if you go to the next one, just to provide actually, a vision of the types and the types of applications we are pursuing using our complete platform. So ENCODE, the ability to design, of course, the synthesis purification process and the delivery technology. I was talking about animal health. And so we have a collaboration that is ongoing with Vaxtera. That's a company in Colombia. The CEO is actually a professor of Wisconsin University, expert in antigen design. And so they designed an antigen, and then we applied our platform. We did the same using a shared antigen, very well known in literature against melanoma. That's a collaboration with the Serum Institute of India. And then I could actually spend the last 2 minutes on the next slide, 16, showing you indeed, how effective, in this case, self-amplified RNA can be. So that's actually a strain that was designed against high pathogenic avian influenza, delivered into a very small study, a proof of concept that we did here in Belgium with our national center, Cienzano, delivering actually the vaccines into 2 doses of, and you see that, we did a little dose range study. So from 0.1 microgram through 0.5 microgram and up to 1 microgram per animal. And so actually this delivered, full protection, 100% survival, no clinical symptoms into the vaccinated animals. And they also survived actually a strong challenge using the highly pathogenic strain. So that's one important result. That was delivered with the dose of 0.1 microgram, which is, basically nothing. And so this is actually, in our hands at least, the very first evidence that mRNA, in this case self-amplified RNA, can be really cost-effective, even for the vaccination of poultry, which is a business for which, cost is everything for the chicken farms actually to be financially sustainable. And on the next one, and it will be my last one, we have now the evidence that when you use actually the same encapsulation chemistry, so the same class of molecules to encapsulate, in this case, mRNA, so this is not SAM, coding for a shared antigen for melanoma. That's also a study we did at the same center here in Belgium. Again, we are not an oncology company, but we want to show that the technologies can deliver functionality. And here again, what you see is that after grafting of the tumors into the animals, when the animals are vaccinated or are given the RNA treatment, so this is what you see on the top right side of the slide. Actually, the survival rate is in the range of 60 to 70%, and similar to the survival rate when the RNA is delivered with the SM102 of Moderna. What you see, in the other chart is that the growth of the tumor is blocked, both when the animals are vaccinated with the Moderna formulation and our calixarin-based formulation. When actually they just receive an anti-PD-1 antibody, there is, a survival rate that is, all the animals actually pass away, and you see a massive growth of the tumors in those populations. So I will stop here. Thanks again for the invitation, and happy to wait the end of the session for the Q&A.
Yes, thank you, Dr. Jose, for your clear explanations about the quantum biosciences, which is, I think, is a reinvent mRNA design, production, and delivery, as well as, I think, it's the most important thing, is also agnostics. And then what I can see from all the 5 speakers, they are enhancing and the collaborations with other partners. So I think we will come to our last speaker. So last but not least, Dr. Chun Lin Sin. So he is the Vice President of the External Innovation from the CanSino Bio. So he will give us in perspective in regards with the AI-driven approaches in mRNA development and manufacturing. I think from Prof. Betty in the beginning, she also used the AI for structuring or to have the vaccine development. So I think I will give the floor to Dr. Chun Lin Sin. So please. Slides? Okay.
Thank you for inviting me here. So I think in this event, I'm the only person from China. So since the mRNA Tech Transfer Hub from WHO and MPP, actually I followed that myself, actually, I have educated in the U.S. and also work in the Novartis in U.S. and China. And back to China 10 years ago, so I joined the Kesehatan to do the vaccine development. So I think I can, in my past 30, 40 years biomedical research experience, I see a lot of from the global point of view and also involve the global health since the pandemic. I personally actually helped the at least 4 countries in the pandemic to do the local vaccine launching in the pandemic. So I see the gap. I clearly see from European, US, and China, and also the RMC countries, including Indonesia as well. I've been have a relationship with Indonesia as a partner actually since the pandemic 2021. So I do have a passion to coming here to show to try to put my foot on your shoes. On your show and see what actually the gap and what is the solution. I agree all of this topic today so far. They put the landscape there, they put the direction there, but I try to share my point of view, what is the solution for that, and how, if we see the gap, how we're going to fill in the gap, and what is the China partnership could be bring you some benefit. So as you can see, now the program start with Hub 1.0 actually from 2021 to 24. So actually it's been the technology access. There have been 15, 16 countries to access the program through the WHO collaboration. Since last year, now they are switched gear to the Hub 2.0. 2.0 actually they're talking about economics, integration, and the sovereignty for each country, for each spoke. Indonesia, of course, is very special because you are the first large of the population and the number 1 large population in the Southeast Asia, ASEAN area. And then you see the ASEAN area, actually they do have also the raising to the biomanufacture leadership, especially they speed up after this pandemic because we all learned the big lessons from the pandemic. They cost us at least 6-7 million people's death life. So everybody learned the lessons from there. As you can see, now Singapore, they do have the niche and the high cost precise mRNA program as well. Vietnam, the government involved as well. And Thailand has very strong research and also the biotech hub as well. So, but Indonesia, with the currently, you're the first one actually, I think, from this hub, government initiate, Minister of Health actually also stand on your point to organize this meeting. I think this is a really good side to key step to make your fast forward. And here, why I think the Indonesia has the strongest foundation in Asia. You see that like we have attending here at Biopharma is a very historically track record. It's a very good company and also WHO PQ global scale partnership and producer as well. And also we have Etana here, very agile innovator, and we do have a collaboration with Etana in the beginning of the pandemic as well. And you see we have a BPOM actually is the MRC only the appear to Singapore. It is one of the highest standard in the ASEAN country as well. Of course, clinical first, we have national KOL, like we have a collaboration with Dr. Alina as well. It's a multiple side of the clinical ability. This morning, you see the sites we have, you have so many clinical trials already. So what is missing here is not the capability, it's the ecosystem integration for the 2.0, yeah. So you see I think why technology transfer is alone is not sustainable. I think the previous few speakers already mentioned that point. The high COGS due to the import, lipid and enzyme. I think Surya also mentioned that point. And the idle GMP capacity, I didn't think you mentioned that, but this is a really, actually in China, we already have this situation. if you quickly build up the capability there, if you don't have a sustainability model to go, you're going to be stuck there. So you really should learn, at least China in the past 2 years, we are going very fast, but we do pay the lessons, yeah. And the cold chain challenge, of course, across the Asia countries, one of the critical important, more important than the rest of the world, actually. Of course, of Africa as well. And then I show the figure to attract your attention, like this is the value of the death. It's really like an economic reality check. If you start everything good, if you don't pay attention about this, only pay attention to tech transfer, what you learn, commercial viability is really, you're going to be die soon somewhere. It's no exceptional. So to post-pandemic, there I see the data, like over 70% of the global mRNA TT project actually stopped due to the commercial non-viability, especially the pandemic requirement coming quick and go fast as well. So all of the facility established from the concept of the pandemic now is facing to awkward situation. So HAB 2.0 priority is to move to low cost and a multiple product platform to see if you can have the successful model. Of course, you already talking about the AI for the cost. I mean, since after pandemic, in the just about less than 2 years, AI is actually impact everybody's life. And then China, we're talking about, if you think about the importance of the AI, of course, you should also study the China model as well. You see, there's one awkward situation I don't want to mention, but this is the fact, like, okay, AI start with US, but it's soon they start. I mean, you already see that the actually fighting between US and China China have always have some way to do something in parallel. So as an option, I think I'm not saying who is good or but it depends on the situation. And I think Indonesia should pay attention for both. And China do have their own AI strategy and the pathway in the future. You will see that at least from Indonesia, you should have the both leverage. Yeah. So, of course, I think Jose also mentioned in their very nice platform, they also mentioned the self-amplify RNA. So this is, I want to emphasize because nobody today in the previous speaker didn't mention that. It is, you see the yellow part I mentioned, I highlighted like a traditional dose actually is a 50 to 100 microgram. But that's talking about the prophylactic vaccine. If you want to switch gear to expand your application to the cancer personalized medicine, we're talking about 1,000 times dose more. You will end up with like a milligram. So it's much more than vaccine. And the other things like nobody mentioned is actually now for the conventional mRNA, actually they have the highly patent owned by UPenn, Joe Weissman. Since the pandemic, he actually, including UPenn, they didn't pursue all of the patent. But sooner in the future, and if you use the conventional mRNA technology, you're going to have to pay attention for the pseudo-uridine modification. This is really heavily patent. I mean, not easy to break through. So, but self-amplify mRNA relatively say you don't have to do the modify. So this is one of the another beauty, like maybe not everybody pay attention. And then of course they can using the much lower dose. It's like at least a 70-80% dose less. Not only the safety is going to be better, it's also the cost of the manufacturer is also the key point. And then this is something enabler, like how we're going to talk about the supply chain sovereignty. So of course, secure upstream ecosystem, do the independent is really the key, like adaptable. The China model, actually, we have an industry park integration. We have local supplier ecosystem, the rapid scale up platform. That's a whole chain from A to Z. We do have the full capacity, including all of building block, enzyme, lipid, all of this. Actually, is which is a much lower cost compared with the US and the European now. Yeah. And then this is I mentioned again, this NAIA is like a nuclear acid industry association. It's actually building, leading by our company in Tianjin area. I welcome you to visit us. Actually, since this association industry park, we can actually, we can get all of the material, lipid, nuclear acid as well, all of the enzyme actually in about minutes or hours rather than days, because they all build in the same area. Yeah. So this is, I mentioned why Kesehatan could be the potentially Indonesia partner. We do have the proof, the track record. We are first with Indonesia in 2021 for the COVID-19 vaccine tech transfer with Biopharma and the University of Indonesia and also the Etana as well. So we are all, I mean, I'm the only invited speaker from China in 2021 for your meeting hosted by Health Minister of Health. Also, we have the joint inhalation TB vaccine trial is ongoing now. We have a HALA compliant process as well. We have a strong alignment with the Biopharma and the Etana as well. So we lead a lot of from our Indonesian, we learn a lot from Indonesian colleague about the HALA, which is one of your very special requirement. And we do have MCV for approval here, PCV tech transfer ongoing with Etana as well. A couple of times we have a site visit from GMP audit from BPOM and MOH as well. So a long-term persist, not the opportunity. So as you can see, the BPOM, of course, is a strategic asset rather than the gatekeeper. So they are mature and trusted, export-ready regulator. So we have worked with the BPOM very smoothly and happy. We build up a lot of experience. This is, because the timing, I will go faster. This is the benchmark. I will leave this slide because this is a lot of information exchanging. Just like across my mind, all of this I prepare in this morning actually. So you'll see this is a list of the different focus on the Singapore, Thailand, Vietnam, Malaysia, and the Philippines, and also what is the good part of the Indonesia as well. So, and the hub, I think the Indonesia hub, the consortium is the model I think we should think about work together, including MOH, BPOM, and Biopharma Etana. Their focus are different, the style is different. And Kesehatan, what we Kesehatan can offer you is a core technology, and then AI also the sampling mRNA IP. We have execution bridging. We have gap analysis. We help you do the global resource scouting, not only including China, integrate China's deep supply chain as well. Of course, Indonesia lead and Kesehatan we can enable for that. So this is my last slide. I do have put here to hope you can add in my WhatsApp as well. Like this is a self-amplified RNA, again, is a sustainable cost, and AI for the speed and efficiency. And like NAVA model is actually for the sovereignty and the supply resilient, of course, plus our 40 years of mRNA technology transfer experience as well. So I do have a lot of more slides. Actually, it's list of the all of this the feature and the thinking what we should do. This including evolve the role from hub. This what is the different? The role gonna be different with today's presentation all from hub 1.0, but in the hub 2.0, their role will be also different, not only yours. And then what Indonesia need to accumulate for the hub 2.0, I list here as well. And of course, the regional collaboration is determined successful. And I think the there's a next step. I think at least a lot of things here. How to make this hub actually work? What is the key point you should really consider? And of course, nobody mentioned, but I think it's important. Nobody can escape where we can get the funding. This is the funding actually, at least from past 3 years, I attending a lot of conference and event. Actually, you might don't even know the name, but there are it exists there, and there are different rules of the applying. So I think it's worth to check all of them if you have a really idea to, but you do need the funding. And this is how you're going to get the funding differently, thinking from last stage to the current stage. And of course, this is the funding strategy. At least I really put here as a reference. And then the channel, and also global, not only including global and China as well, and of course AI. So you can see, you can have this slide, and really have the information changing. So I think I stop here. We can have some discussion. Yeah.
Yes, thank you, Dr. Chun Lin. I think it's really comprehensive presentations that you already analyzed the Indonesian situation, and also not only finding the gaps, but also give us several options for to fill in the gap. Okay, so I think we are moving to the next session, which is the questions and answers. So I believe, like the participants here already have questions. We have 6 speakers. Professor Betty from the academicians, and also we have Christian from the industries that doing the mRNA innovations, and Dr. Indri, which is from the biopharma, the manufacturers. Also online we have Mr. Rajinder from the DCVMN, as well as Dr. Jose from the Quantum Biosciences that provide the platforms, and the last but not least is Dr. Chun Lin from the Casino Biologic. I open the floor for questions. Anyone? Okay, silakan, Dr. Bina.
Thank you for the opportunity. My name is Dr. Bina from Fatmawati Hospital. I would like to ask for Prof. Betty, and you have mentioned about the dengue research, and for now the dengue vaccine is available only for 6 years up to 45 years. Is there any further research for the safety for the children below 6 years, especially for infants? And the second question for Etana. Etana has produced the vaccine PCV13. Is there any further research for the PCV21? Thank you.
Ya, silakan, Prof. Betty.
So thank you very much for the question. So actually the vaccine that is already a commercial one in here is still have some limitation. But right now in the clinical trial from the MSD, I know that they all also goes to the children under 5. So I didn't get any result from that. But I hope that this will be better than 2 previous vaccines that already commercial in Indonesia. And then what we designed for the vaccine from our side, that we using the mRNA technology, we hope that it can be work for the children under 5 also, because we're thinking that the severe cases is right now, even though it's already moving to the adult, that you know that the vaccine is for to prevent of the severe disease. So even though right now it's like moving to the adult also for the severe cases of dengue, but actually it's for the children under 5 is still quite dangerous. So we hope that our vaccine can go to this in the early age of the children. So is it answer your question? I hope.
Ya, sudah menjawab, Dok? Iya. Silakan, ada pertanyaan lanjutan atau cukup, Dr. Bina, untuk Prof. Betty? Cukup, oke. Silakan, Pak Christian.
Yeah, so for sure for PCV more than 13, I mean the valence itself, it in our mind. However, we should also thinking that it's not that simple. I mean, it is also related with the cost and also later on related with the demand itself. I mean, the current situation is PCV 13, right? However, in the future, we really need PCV, for example, 21, Valent, and how it will work. What will be our business model later on? Would we, for example, import first and then later on we will produce in our facility, or maybe no need to produce in our facility for PCV, for example, 21 or maybe 2030, 23, for examples related with the valence itself, related with the strain itself, whether the strain it is a lot of available here in Indonesia. So it is part of how we should think about the cost, the needed of the society also, and then later on the business model that we should thinking. I do believe that PCV with the higher valence is still also keep in our mind, but we still thinking about these three, whether it is part of our pipeline that we should going on, and then when we should launch this project if our calculation related with the business, related with the demand, also related with the cost later on can meet with between us and also with the others supporting related. Is it answer your question? Yeah, thank you.
Okay, so is there any more questions from the audience? Yeah, okay. The first one is at the front and then Mbak Falah the next.
Yeah, thank you. Let me to introduce myself. My name is Endang from PT Biotis Biotechnology, Biotis Pharmaceuticals Indonesia. I have question for Dr. Jose. Based on your experience using the intensify MIDI to develop the RNA, I think it is a short time to produce with the modern technology compared to the old technology for mRNA production. But we have many challenges, I think. The first one is the yield of RNA when we produce from MIDI to the upscale, and also the dsRNA or impurity, and also the LNP. And I think for residual RNA also is very high. Have you thinking about how to overcome when we using the MIDI to the upscale? It can use MIDI every time, or we need to another technology or MAXI for industry? Thank you.
Okay, so Dr. Jose, can you understand the question well? Okay, good. So
Yeah,
please.
yes,
Yeah.
absolutely. It was very clear. Thank you. No, indeed. So that's a very good question, and indeed the way we designed our technology, as I was saying, it's based on a process. The process has been made agnostic to allow actually the same conditions in terms of temperature, time, kinetics, and all the compositions to be the same from milligram to kilogram scale. And today, indeed, you are totally right. When our partners use our Intensify MIDI, which is the system that we deliver to produce GMP material, today actually almost everybody is using it at the gram scale. Actually, one of the WHO, actually not partners, but Afrigen, actually, that also Dr. Suri referred to, actually they did 2 days ago the very first batch at the largest small scale, which is actually 5 grams, but the system is designed to produce 24/7. Then it's a matter actually of us to understand what our partners desire.
Okay.
Use mRNA MIDI is almost over-designed. If they use self-amplified RNA for poultry vaccination, what we have is very big because it delivers 100 million doses per day. And then it's really a matter of indications. If you tell me about, we mentioned today about dengue vaccines. If dengue is based on mRNA and the dose is, 25 micrograms, and the goal is to produce 50 million doses per year, then we will go from the MIDI to the maxi. And so the system is designed to, to address those needs. But of course, today, as a technology provider, what we cannot do is to anticipate what people will need, because otherwise I would have like 3 other machines. So the fundamental machine is there, and then depending on the application, we'll come with upscaling or additional flexibility, and this is something that will come next year. So this is from the production and purification perspective. Now, indeed, you mentioned encapsulation. So Chulin mentioned, also referred to IP, but also to the use of methylated pseudouridines. But so if I answer on the encapsulation, our goal actually was to deliver freedom to operate. I think we are there. And for the rest, today we have the evidence that the LNPs deliver in terms of functionality, immune response, or anti-cancer immune response. And we are actually developing a process that will allow to go to kilograms actually of encapsulated RNA per year. So scalability is really, a fundamental piece of the way the technology has been designed.
Okay, does that answer your questions? Okay. So thank you for your answers, Dr. Jose. So we will move to the next questions. Mbak Fala.
Thank you. My name is Fala and I'm from the MOH. I would like to ask Dr. Chulin Sin. Thank you for your presentation. You mentioned earlier that ecosystem logic matters more than technology transfer. Could you please explain more on that, since I think maybe, especially in LMIC, the technology transfer itself is the starting point for the ecosystem building. So could you please kindly explain more on that? Thank you.
Yes, thank you for the question. My understanding is like tech transfer can be tried to standardize, just like exactly what we do, what the WHO do for the hub of 1.0 program. They actually collaborate with also quantum to using all of this fancy, the standard. Everything can standardize, try to using the module version. So this is actually can help really the successful for the tech transfer. But the ecosystem is talking about just like the tech transfer can be treated as a, the training school, training program, and you as a student, as a spoke. So you learn everything there. You might, like, for example, I think the from the hub's 1.0 program, actually Biopharma Indonesia actually is one of the top students. You graduate now, but now you are in the phase 2. It's actually you're going to have to survival by yourself. But for the survival, whatever you learn standardized from the tech transfer is not enough for your survival. Of course, the different country have a different situation. I would give you example, like we are actually starting quite similar timing. Most of the China's mRNA technology platform or company also start with like early of 2021. And then after 3 years, you have 16 spoke for the WHO in our middle income country, and then maybe 4 or 5 of them is actually have a very top records, graduate. But in China, in the similar years in parallel, we have more than 50 established mRNA-related company. So what is the difference is because the China ecosystem is already reaching is there. So now if you think you are entering the hub 2.0 phase, I would thinking the China can claim we're in the phase 3.0. And what I mean is like not only we have more than 50 established, we have they all, at least half of them have the GMP facility. And we have at least 40 of the, the IND stage mRNA the product, including not only the vaccine, also it already expand to the therapeutic field. And I think nobody talking about the recent 2 to 3 years, 2 to 3 months ago, there is in the field, it's like everybody talking about in vivo CAR T. This is another brilliant opportunity actually can bring the mRNA technology to be viable. So that at least I think from if you in the 2.0 phase, of course it's a little early. The reason is like because the ecosystem. Of course, your starting point is kind of, you're gonna have to set the phase. I think in the maybe next year or two, you have talking about only one center, for example, like using biopharma as a center, and then you set up the successful case. You might just like at least have the scale up GMP ready, and also maybe IND ready. That is something. You set up the one case to go, and then the 2 years later, you will not only for the one case. For example, how are you going to expand to the quickly to the 50 case to make your own country really sustainable? That is the have to be considered ecosystem. So that's why when I present, I'm not only talking about what the technology we have, and I also mentioned we do have established a hub for the nuclear acid industry association. In the bio industry park, we have more than 50 different companies. They all actually align in the same tech park. They can supply you with a lipid, with a nucleic acid, with all the building block, and including also the AI design and the logistic as well. So that's why I'm talking about the ecosystem. With that, you can really survive. Yeah, thanks.
Yes, thank you, Dr. Chunli. I think it's really clear what the integrated ecosystem is. I think it's really easy to say, but it's not that easy to be done, right? And I think here, everybody from the several stakeholders that involving in the vaccine development are here. So I think it's time for us to really work together and then build a concrete actions, what we're gonna do next, and then what will be following up of this meeting. So this is not only the meeting we meet, without a concrete actions. I think that's the point of this meeting. Okay, so we still have around 10 minutes for the question and answers. Silakan, Prof.
Thank you, Bu Indri. I'm Desi from ITB. So we have actually developed several vaccine at ITB. So during the COVID-19 pandemic, so we joined the Vaksin Merah Putih Consortium. So we developed the adenovirus-based vaccine and also protein subunit-based vaccine. And during the last 2 years, we also started to work with the mRNA vaccine. So we have developed the hepatitis B mRNA vaccine and also the dengue vaccine. For the dengue vaccine, we have the full length and also the multi-subunit vaccine candidates. So what I want to ask, because following what Bu Indri just stated, we need to have a real action. So I wanted in order to advancing our vaccine candidate, so I want to ask what opportunity that exists for us to collaborate with Etana or with Biopharma. Okay, and also I want to ask maybe to Bu Indri, so what can the Ministry of Health can facilitate this collaboration? Thank you.
Okay, thank you, Prof. Desi. Okay, so I think Prof. Desi is a representative from researchers, from the academicians, that she's already done several projects, several studies in regard with the developments of the vaccines, like several vaccines that she mentioned. So she wants to ask, what should I do next? So whom I can have a collaboration with. So because I think, you know, since the beginning, like all the speakers saying about collaborations, about the academician, business, governments, you know, in one line, but like actually, this is like a concrete questions from some of the researchers. So, what should she do next, and what can you provide to her? So, like her research can be implemented and can be continue to the next phase. So I think this is like a question for all the speakers because I think it's related to all the speakers, the question. So I think I will start with Dr. Rajaran from the DCVMN. So could you please ask the questions and then follow up with the Dr. Jose, and then later on with the speakers here that's already here present offline. Please.
Thank you very much once again. See, as far as collaboration is concerned, I think most welcome. Almost everybody is here in this room basically to collaborate with one another to find solutions to the common problems that all of us have. So I would be very happy to welcome any suggestions and inputs from all the members, whether you are from academia, industry, or government or regulator. I think this is the opportunity, you know, this kind of platform that are being hosted by Government of Indonesia and Department of Pharmaceuticals that bring people together, the like-minded people. That is what is my new message is LMP, the like-minded people who have to come together and forge collaborations to make sure that we are moving in the right direction with a common objective, common goal, and a common strategy. I stay at your disposal.
Okay, thank you, Dr. Rajaran, for your answers. I think we will move to Dr. Jose.
Sure, thank you. So indeed, and it was actually a piece of my introduction in my presentation today, is that this is something we experience. People in front of us want to work together. So I think that there is several things to consider. From my perspective, the very first, coming back to the tech, because the tech is what we use to develop something which is actually the real goal, which is a medical benefit for the society. There is two things, and I think that Dr. Suri mentioned that it's funding. So at some point, someone needs to pay, and the one that will pay must see actually the benefit. And so I think that probably and I know that in the WHO hub, this is something that comes on a regular basis, which is actually, what will be the benefit? What should we prioritize in terms of development? So this is probably the very first thing. The beauty, I think, of RNA, and I'm saying that as a chemical engineer, actually, the beauty of RNA is that, and my first career was in cell culture and viral vaccines and viral vectors. The fundamental difference between that type of biomanufacturing and RNA is that RNA can be platformized. So it can be extremely efficient in terms of process development and product development. So I think that we have a beautiful case in front of us where you can minimize the cost and maximize the output, which is, at the end of the day, the societal benefit for all the countries that want to invest. So this is, I think, the very first thing is actually what do we want to achieve and what is the priority list for which actually RNA can be a very interesting modality to achieve our goals. And then the second thing is about, and I think that Julien made a very good job at saying that, is he mentioned several times the term ecosystem. So at the end, indeed, one thing is to develop something that works. The other thing is to make sure that this, achieves the goal, which is to reach the patients. So it's all about commercialization and making it financially sustainable. And here, actually, I can only talk, But so, we are a small actor as a technology provider, but indeed, so we need actually, the most relevant stakeholders in each country, in Indonesia, more specifically today, to make sure that all this money invested in development will really reach the benefit for the country. And I think that RNA has the capability of providing a great benefit and decrease the total cost that the country is putting in health. So that would be my contribution to this debate.
Yes, thank you. I think from both speakers, it's clearly stated that first we need to know the priority. Here is the priority from the Ministry of Health. I think we have, 14 antigens? That is our priority for the next 5 years. So if the researcher can follow those priorities, that will be good. And the second one is that from the perspective of those both speakers saying about how this can be a scaling up to a manufacturing process, because and also the involvement of the regulations is regulators since the beginning. Because mostly the researcher, they do the research, but I mean in the end of the day, if we want our research to be implemented, then we should also follow the requirements for the regulators, which is I think is the BPOM. The BPOM is not here, so we can ask them later. So so I think I will give the floor to Prof. Betty as one of the actors as well, one of the researchers. So from the experience, what are the problems that you face, like Professor Desi faced now that you have this, so I already researched this, so what should I do next? I mean, like, does my research is qualified enough? And then if not, what I should do? And then what can be done so my research it can be elevated into the next level?
Okay, so because we are the same in the academic side, so I'm thinking that collaboration is the most important. So if you want to join with us, I was really happy because I also conducted for the dengue research, for the dengue vaccine. So I'm thinking that when so many academy doing the same thing, it's like overlap each other, spend time. So I think together will make us stronger. I'm thinking of that. And then of course, beside that, because in our project also, I include all of the researchers that interested on the dengue vaccine development. Please join with us. And I'm really sure that it will be accelerate of the dengue vaccine development. And I also join with the collaboration with the Tsinghua University that have experience to doing research on the vaccine and also move to the industry. And this is the luck of our team, because we always maybe good in the lab scale, but when we go to the industry, it's really need collaboration with other university, and also join with together with the industry in the beginning when we design the vaccine, because it will help that easily to move to the industry to produce of the vaccine. So I think this one together make us stronger, I'm thinking.
I agree. Together make us stronger. Yes. I think that is also the responsibility from the Ministry of Health. Yeah, Ibu Eni, coba tolong Ibu Eni. Ibu Eni. Yeah, that is our job in the Ministry of Health as an enabler. So because we have this like really good researchers that research the same dengue vaccine, for example. So how we can collaborate them so they can produce the vaccine that is really needed by us in the Ministry of Health. And I think we go to the manufacturers. So I think I will go to the biopharma first, because like the biopharma now, they conduct several research as well, and then they are really close with ITB. Yeah, so I would like to ask you, so how Professor Desi can move forward with her research? Please, Bu.
Thank you, Bu Indri. Actually, yes, biopharma have a lot of collaboration with the university. For example, like when we are developing MERS mRNA, actually when we are make the antigen for the MERS, we also collaborate with Pajajaran University. So a lot of that we are developing the vaccine, we get the insight from the university. For example, like hepatitis B. Yes, hepatitis B is included in the Ministry of Health 14 antigen. And then after that, there is the new vaccine will be issued by Ministry of Health is hexavalent, whereas hepatitis B add with another antigen. We need to discuss what is the benefit of this hepatitis B into mRNA platform, because Bio Farma already have hepatitis B with the protein recombinant platform. Actually, we are open for the collaboration with, because we make together, even with Etana, we also make together to make a stronger, not to be competitive each other. Thank you. Actually, together is important for accelerate time to market also.
Okay, so Prof. Desi, you can contact Ibu Indri ya for her promise ya. So, so you can have like further collaborations. Okay, the last two speakers. So from Etana, please.
Yeah, so thank you. So first of all, this morning I just say hi to I just say hi to Ibu to Ibu Erna, just to follow up what is our next action. And then I just realized that Ibu Erna, oh, I also go to the Ministry of Health in the Kuningan. Oh, then we meet, then we should discuss what the next project that we will work together.
That's good.
Yeah, that's the first step. Just say hi, okay? Then Itana, we work as a system incubators and also system integrators, which is the one that we already conduct with the University of Indonesia, how we accelerate the research to increase their TRL faster. So we channeling between University of Indonesia and Tsinghua University to accelerate the research. And then after that, what we can do, for example, the Ministry of Health as the government, we always think that, okay, the funding maybe should come from the university, from the government or from the industry. However, I just thinking that a small step, just like the government of Indonesia, the Ministry of Health knows that there is a funding which is open, and there is a statement and call to all of the university. It is a small step, but it can help a lot. Maybe one of the researcher can apply and can get the funding. Maybe from the Ministry of Health can make a statement that this is part of their priority disease, and then it will be linked between the priority disease of the Ministry of Health and then the disease, or maybe the project that going on the university, right? So it is a small step, but it can help a lot to the researcher, and also it can help a lot also to the industry. So for example, I sometimes said, okay, we know there is a funding in this university, but they need a lot of collaboration between two universities, and then we invite, and then we should select whether it is interested or it is have the same common intent. So what we do is connecting each other. So it is just a small step, but it helps a lot to make the ecosystem like Dr. Sin just mentioned. So ecosystem is also important.
Thank you. Okay, last but not least, Dr. Chen Lin, please.
That reminds me, I think when the first pandemic hit us in China, we're the first group that can access the sequence. But in that time, it's actually echo my first question. mRNA, I just start. I using the NEB enzyme and using the sigma lipid. So for all research grade, I think I'm the first person in the whole world actually developed this mRNA COVID-19 vaccine in about months. We do have the material, but its stability is it's only 1 week. And then the cost actually is $100 per dose, per mouse dose. So we think this is a proof of concept, just like a SARS the SARS-1. And maybe a couple of months it's going to be gone. But unfortunately, this is a real disease actually spreading very fast. We have to deliver the vaccine to apply to real people. So that's why we have to give up for the mRNA because we do not have the ecosystem to support us to launching the mRNA-based vaccine. So we switched to adenovirus vector-based because the only reason because we not believe like viral vector is better is because the ecosystem. We have all the material to deliver the vaccine using the vector virus-based system. So that's why, like, okay, ecosystem also important. For you, for example, if you think you do have the very good candidate vaccine, they want to connect to the next step. And of course, you're going to check with all of the surrounding partner if they have the ecosystem support you. If do not have, and then of course you're going to have to launching that product through the collaboration with outside of the country as well. So the other thing I want to remind you is like, so if you want to publish, or we only want to generate IP, that's free to go. That's another angle. But if you do in seriously in your heart, you want to develop the vaccine as a product, and then you better, when you start this project, you better have the collaboration with the company. Yeah, because they have, there's two desks. One is like a regulatory. What is their concern? Be a product to put the human being. The second one is like a CMC part. That's usually all of the research, they do not have ever think about the CMC part. It's quite difficult, but also it's a challenge. So if you have a good product, maybe you can't go through the CMC part. It's going to be no tomorrow. Yeah. So it's better talk with discuss with the company if you really want to launch the product. Of course, obviously, if you have the antigen finding, then which deliver system put this antigen to generate more robust vaccine effective. That's another thing. Actually, usually the research cannot answer that. It's going to have to, like biopharma, they might have a lot of case to reference to can help discuss with that. So I think if you really want to launch the product, it's involve the industry as early as you can. Thank you.
Thank you. Okay, I think we are coming to an end of this session. So I would like to thank to all the speakers. So Prof. Betty, Pak Christian, Ibu Indri, Dr. Chun Lin, and also online, Mr. Rajrans, and also Dr. Castillo. Thank you very much for your times. So I think for the last 2.5 hours or 2 hours, yes, for the last 2 hours, we are listening about collaborations, scalability, and also gap and challenges that being faced by not only by the researchers but also by the manufacturers and also by the technology platforms. So I think now it's the time for us to fill in the gap that is already mentioned by all the speakers. And then also I would like to acknowledge that may today's knowledge is not only stay in this room, but continue to grow into concrete actions and innovations across our institutions. Innovation is not just creating something new, but it's about creating something meaningful. When science, collaborations, and purpose come together, even the impossible become achievable. So thank you very much. For the last thing, I would like to close with pantun. So pantun is like a rhymes, but in Indonesia, yeah. I'm sorry I cannot do this in English because it's gonna be a little bit weird. So I'll do it in bahasa, ya. Oke, pergi ke hutan membawa pelita. Asik, aduh. Oke, pelita redup ditambah api. Ilmu dan inovasi kita bawa bersama agar kesehatan Indonesia makin maju dan mandiri. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Saya kembalikan kepada MC.
Okay, ladies and gentlemen, once again please give it a round of applause to all speaker and moderator. And thank you very much to moderator and all speaker for the second session. We kindly to all speaker and moderator to stay on the stage. to give appreciation after this. Saya mohon Bapak Ibu untuk tetap berada di atas panggung untuk pemberian apresiasi. Dan I would like to invite Mrs. Weni Indriasari to come to the stage to give appreciation to our speaker and moderator for the second session today. Oke, terima kasih banyak. 3, 2, 1. And next, silakan. Terima kasih banyak Ibu. Kita foto dulu ya Bu. Oke, 3, 2, 1. Oke, dan selanjutnya sekali lagi kami mengucapkan terima kasih untuk narasumber kita di diskusi panel sesi kedua ini. Oke, 3, 2, 1. Ya, dan terakhir, terima kasih untuk moderator kita. 3, 2, 1. Oke, mungkin setelah ini kita foto bersama ya. Let's taking picture together. Bapak Ibu, kami persilakan. Kita foto bersama terlebih dahulu sebelum meninggalkan stage. Alright. 3, 2, 1. Oke, terima kasih banyak Bapak Ibu. Kami persilakan untuk dapat kembali ke tempat. Thank you very much. Please kindly return to your seat. Baiklah, hadirin yang kami hormati. Dengan ini usai sudah rangkaian acara Seminar mRNA Innovation to Implementation Integrating Science, Regulation and Global Collaboration. And we believe this initiative will enhance knowledge and capacity of our researchers and achieve betterment of our generation. So ladies and gentlemen, with the end of the ceremony, I am Bella Edarwan. We have the organizing committee extending our appreciation to each and every one of your attending. Thank you so much for your participation. Dan akhir kata, izinkanlah kami pamit undur diri. Dan mohon maaf apabila ada suatu hal yang kurang berkenan di hati Bapak Ibu semuanya. Indonesia sehat, Indonesia kuat, Indonesia maju untuk menuju Indonesia emas. Wassalamualaikum warahmatullahi wabarakatuh. Selamat sore dan sampai jumpa. Baik Bapak Ibu, untuk Bapak Ibu yang ingin materi ya, paparan materi tadi silakan untuk men-scan barcode yang ada di layar.